Sarni, you're muted.
Good morning, everybody. How are you guys doing? Welcome to day 2 of the RISE Symposium. I'm going to be the host of this particular show. We have 22 lovely presenters today, and this is the part of the program where nobody gets judged. All you guys get to stay home and be winners. So looking forward to the culmination of your last 8 weeks of research in the RISE Symposium. And we're going to start off with an introduction video of today's speakers that were given today. All right, Marissa, thank you very much. And we'll go straight into our intro videos for all the presenters.
Hi, my name is Tiffany Larabee. I am an environmental science major. I am transferring as a junior and I currently live in Fontana, California.
Hello, my name is Martha Lopez. I'm a biology major and I'm a transfer student. I currently live here in LA with my roommates. I call them mom and dad.
Hi, my name is John Pinello. I'm a microbiology major and this is my 3rd year of college. I currently live in the Antelope Valley.
Hello, my name is Ethan Sirlard. I'm a 2nd year biology major and I live in Los Angeles.
Hi, my name is Kendrick Davis. I'm a 4th year bioengineering student and I live in Moreno Valley.
Hey, my name is Alex Rojas. I'm a 3rd year microbiology major and I live in Victorville, California. And here's my dog.
Hey, my name is Jesus Camba. I'm from San Francisco, California, and I'm a microbiology major starting this fall.
Hi, my name is Alexandra Richardson. I'm a statistics major coming in as a 3rd year transfer to UC Riverside, and I'm from Elk Grove, California.
Hello, my name is Nicole Schad. I am currently a 3rd year transfer and a biology major, and I am in Riverside right now.
My name is Radhika Amin. I'm a 2nd year neuroscience major, and I live in Fremont, California.
Hi everyone, I'm Michaela Henry. I'm a 3rd year transfer student. I'm a math major and I live in San Gabriel, California.
Hello, my name is Devante Kane. I'm double majoring in math and physics at UCR and I'm currently living in the city of Banning.
Hi everyone, I'm Natalie. I'm a 3rd year neuroscience major and I live in Fontana.
Hey everyone, my name is Sarah Kavar. I'm a 4th year neuroscience major and I am from Corona.
Hi, my name is Daniel Avalos. I'm an undergraduate transfer student majoring in biology, and I'm from the Riverside County.
Hi, my name is Michael Asaf. I'm a neuroscience major, and I'm transferring this year to UCR. I also live in Riverside.
Hi, I'm Fatima Macedo. I'm a 3rd year biology major, and I'm from San Marcos.
Hi, my name is Stephen Johnston. I'm a 3rd year studying cell molecular developmental biology, and I'm living in Riverside, California.
I'm Gabriel Chavez. I'm from Paris, California, and I am a 2nd year biology major at UCR.
Hi, my name is Stephanie Loyola. I'm a 3rd year incoming transfer student at UCR. My major is biology and I live in Riverside, California.
Hi everyone, my name is Shakira Palmer. I'm a 3rd year biology major transferring to UCR. I live in Riverside, California.
Hi, Josh here from Riverside, studying math and physics, and hopefully, with any luck, I'll be graduating this coming spring.
Okay, well, great videos. It was great seeing everybody, and everyone had some very creative presentations. So we're going to move on to our first student speaker of the day. These are students who were nominated and volunteered to just share their particular experiences with RISE and how learn more about their particular stories, as well as how RISE has impacted their experience for the summer. So our first speaker will be Mr. Alex Rojas. Thank you very much.
Thank you, Nee. Hello and good morning, everyone. I hope you all are doing very well and staying safe by wearing masks, practicing social distancing, and all that good stuff. My name is Alex Rojas, as Nee mentioned. I am a microbiology major, transferring from Victor Valley College, and I'm very excited to start fall classes next week. Now, I know this remote and digital experience may not be ideal for some people or what they might have hoped for, but I do want to make it clear that everyone, from the students to the faculty and the RISE coordinators, everyone's been working really hard to make the best out of this research experience. I am a part of the Summer Bridge to Research program, and so far, my personal experience has just been phenomenal. I worked in Dr. Jinsong Zhang's lab along with Dr. Sun to observe changes in California air quality during the COVID-19 pandemic. I am more than happy to help in research with any sort of topics or problems to better our understanding of the sciences so that we can make the world just a better place to live in. And you know, the great thing about the STEM field is that a lot of topics can be interdisciplinary, allowing for the opportunity of exposure to a variety of subjects. This program gives us the chance to broaden our horizons and perform undergraduate research, maybe even in a field we never really thought of. At any rate, the program was extremely helpful and gave me the experience to work on a research project from start to finish. I had the opportunity of beginning with an idea, working out a plan to support my hypothesis, and doing the research on my own. I collected a massive amount of data and used various programs to analyze it and make the appropriate calculations. I learned how to use a new data analysis software to generate graphs and trend curves, although it was a little tricky to figure out at first. Working on this, it really helped to build my confidence level, especially now knowing that I can accomplish this magnitude of research almost entirely on my own. Almost entirely. I did communicate quite a bit with the professor and mentors as we collaborated on the compiled data, trying various ways to present it in neat, easy to understand graphs. In doing this, I also realized the challenges I faced when working on this project, specifically trying to find the best method of averaging and presenting the data. I realized the courses I would need to take, like statistics, to benefit my academic goals and prepare me to succeed in future careers. This collaboration with faculty mentors, although a little intimidating at first, was nice since I felt like I was also a grad student or even an employee in a research and development career. I felt like a professional as we communicated back and forth. In general, my experience helped me develop crucial life skills like effective communication, teamwork, collaboration, and problem solving, to name a few, all of which I can definitely use in my future career. While I'm sure many students, myself included, would have loved to run experiments physically in a lab and collaborate with others in person, this remote setting instead gave us the opportunity to work from home. Now, I know this work from home might have been more stressful for some, but I personally did enjoy it. I mean, I was able to research, collect data, perform calculations, and even collaborate with others, all remotely from the comfort of my own home, like right now. A variety of workshops was also offered through the program that cover topics like career and network building to applying for graduate or even medical school, to even studying abroad, which I had not really considered before. These informational workshops were really helpful and taught me more on how to present my findings professionally and effectively, how to write research papers, and just overall how to better my academic career here at UCR. Each of these workshops and meetings held throughout the program not only gave valuable information, but they were also really engaging and fun at times, especially with those Kahoot games. It kept everyone on their toes as we all participated together during these Zoom meetings. Also, talking with fellow students and peer mentors was nice as we all had fun bonding, networking, and sharing our experiences together. From workshops to researching to collaborating with peers and faculty members, this program taught me many, many things, and I'm just so happy that I've been part of this amazing summer program. Thank you all for listening and being here, and I look forward to hearing all the presentations today, and I wish everyone the best of luck.
All right, thank you very much, Alex. What a great way to start off our program. And I definitely appreciate the ability to stay home, and you look very, very comfy as well. So thank you very much again. So now we're going to have our second speaker of the morning, Ms. Radhika Amin. You could take it away. Thank you.
Okay. Hello, everyone. My name is Radhika Amin. I'm a second-year neuroscience major here at UCR in the College of CNAS. And since last fall, I have been a research assistant at the UCR Brain Game Center. So I joined this lab because I wanted to know more about cognitive impairments and how those functions may be affected in individuals who are cognitively impaired. And this interest kind of stemmed from my experience with concussions from when I played soccer in high school. So specifically during RISE, I worked with my PI and coordinators at the UCR Brain Center to develop a cognitive battery to assess the functioning in stroke patients, which is something I came into college dreaming of doing, like working with individuals who suffer from these types of conditions. So when I first came to UCR, I thought that once I started school, I would soon know what career I would want to have. But deciding what you want to do in the future is difficult for any undergraduate student, no matter your age or background. And I think that RISE has given me a glimpse into the vast career possibilities within various fields. There's so many intricate parts of molecules, the environment, and even ourselves that I learned about because of RISE. I didn't know that organisms could change their entire structure because of environmental factors, or that gut-brain interactions could play a role in our body's response to COVID-19. I think that hearing about the research of others has not only been extremely interesting, but has also led me to become a more well-rounded student. I have now realized the connection between all of these interdisciplinary studies, as Alex mentioned, and I can apply what I have learned to my own research. At first, when the program started, I was a bit worried that I wouldn't have any concrete data to share with my peers at this symposium. As the program progressed, I realized that everyone was at different stages in their research. No matter how much progress I had made, it was okay. It wasn't the progress that mattered, it was the process that mattered. I got to experience this firsthand through the many drafts and revisions of my stroke battery proposal in my particular research project. I learned to enjoy every step of this journey because along the way, I gained so much knowledge. And without the support of my PI and lab coordinators, this wouldn't be possible. So I'm very thankful for all the opportunities that RISE has provided. Research is a complicated process, and working with experts across many fields has shown me that patience and being steadfast is crucial. And especially during this time when I thought it was impossible to meet and interact with new people, I got to do exactly just that. So I'm grateful to have gained a greater understanding of the research I am conducting and also the research of my peers. When I joined my lab last year, I couldn't have imagined all of the progress I have made thus far. The past 10 weeks have definitely contributed to the career path I will take in the future, and I am excited to see what the future holds for all of us RISE participants. Thank you so much for letting me participate in this amazing program, and I hope everyone enjoys the program today.
Thank you very much, Radhika. That was extremely inspiring and yeah, very, very heartfelt as well. All right, so we will have our last speaker of the morning, Ms. Natalie Tajeda. Thank you very much for turning on your camera and logging in. And the floor is yours. Take it away.
Good morning, everyone. I'm Natalie Tajeda. I'm an incoming third year. I'm a neuroscience major, and I'm part of the PERSIST cohort. So PERSIST is a sophomore year-long program that provides mentorship and support to students interested in research or biotechnology or any STEM-related fields. So I joined the program. Initially, I wasn't going to apply to the program because being a first-generation Latina, being a woman in science, it has been challenging, and I didn't think I had what it takes to be a researcher. I didn't think I had the resources or the skills, or I didn't even think I was smart enough to be a scientist. And I was filled with a lot of self-doubt. And the more I thought about it, the more I thought that I can't let my fear of rejection or my fear of failure stop me from achieving great things or having new exposures to new opportunities. So I went ahead and I applied. And I remember when I got in, I was in chemistry lecture, and I had read the notification that said, "Congratulations." And in that moment, I was overwhelmed with happiness. And I had left class and I called my mom immediately because my mom is my #1 supporter. And I told her that I'm one step closer to achieving our dreams. I'm doing everything for her. And I know that she was so proud of me because I'm one step closer to being someone in life. And I'm so proud that I got this opportunity to become a scientist, to experience research. And now, once I started PERSIST, we were a bunch of small little sophomores, all very shy. I remember we were practicing our elevator speeches with each other, and we were all shy and timid, but then we had to get out of our comfort zone quickly because we had to experience and interact with a lot of the faculty members. And one of our main events was Escape which is a networking conference where we kind of like speed dated faculty because we had to constantly switch tables and interact with them. So we constantly had to grow and we had to get out of our shells quickly. And now some of us are getting ready to present at our symposium, and some of us already presented. So seeing the personal growth through my Persist group has been an incredible journey because we've all been through this research this whole year together, and it's been amazing being able to witness how much we have all grown. Now for RISE, I really want to thank Nia and Gina and all the peer mentors because they created a welcoming and a supportive environment. Being online is challenging as it is, but they did an amazing job in guiding us through the whole research process every step of the way. Every weekly meeting was something that I looked forward to because I was always and it was incredible to learn new resources. I learned about professionalism, UCR, the Career Center, lots of personal growth as well. One of my favorite talks was about studying abroad because I didn't think science majors had the option to study abroad. So it opened my eyes to new opportunities and that I am now ready to take on one day. And now I also want to thank Dr. Sue Wessler and Dr. Connie Nugent because these are two empowering women that are constantly breaking boundaries, and I strive to be like them one day because through them I realized that faculty and professors are human beings too. I learned how to interact with them well. We had conversations from COVID to politics to humanitarian issues. So being exposed to this environment with them really helped me see that they're human beings too. And there might be a gap between our knowledge, but they're here to help us learn. So I'm really appreciative for them and for their guidance. And it's been an incredible journey to see how much we have all grown. My professionalism has grown immensely throughout this opportunity as well. I want to become a physician someday, and all the skills I learned through RISE and my lab as well has taught me that I can be someone someday. I can, with the right guidance and the right mentors, I can be someone someday. And this is what RISE has taught me, that I can grow out of my comfort zone, that I can strive to be whatever I want to be without stereotypes or boundaries. I am my limit, you know? And it's been incredible to see how much I have grown throughout this entire year. And lastly, I want to thank all the members who put this program together because we wouldn't have been able to grow as much without all of you guys. And some advice I would like to give to any future RISE participants is being a scientist means we're constantly growing, we're constantly learning, and the more we learn, the more enthusiastic and more curious we become. So be a scientist, be brave, and don't be afraid. Enjoy your journey. Thank you, everyone.
Thank you very much, Natalie. That was amazing. And again, I feel all your sentiments. Sometimes, yeah, we always need a reminder that faculty are in fact human. And of course, it's really about, the process and learning and so many things that we've put together just for you guys. So just to kind of give it back, we, as in myself, Marissa, all the faculty, we are here for you guys, for the students, and to make this experience as best we can. So we will be moving on to our first speaker, and just to kind of give a brief breakdown of how the presentation and symposium will proceed. Our oral presentations are scheduled for 12 minutes each, and each student will be presenting for approximately 10 minutes with 2 to 3 minutes of questions at the end. And all guests, attendees, you are welcome and encouraged to ask questions via typing in the webinar chat, or you can raise your virtual hand on the participant panel. If you see how to raise a hand there, you have the option to answer live. We will unmute you, so you can speak directly to the panelists or to the presenters, and they can respond via live. So without further ado, we will move on to our first speaker of the morning, Ms. Tiffany Larabee. And if you could turn on your camera whenever you get a chance. Thank you very much. All right, so your presentation is loaded. Marissa is hiding behind the green curtain supporting you. Actually, all of us are supporting you. So the floor is yours. Please take it away.
All right. Hello, my name is Tiffany Larabee. I am an environmental science major working under Dr. Francesca Hopkins. I'll be presenting on oil and gas infrastructure mapping for methane emissions in Kern County, California. Next slide. So here's just a quick overview of my presentation, as well as a methane leak in infrared. Next, please. So some background information on methane. Methane is an incredibly potent greenhouse gas. While it has a shorter lifespan than carbon dioxide, the most well-known greenhouse gas, it traps heat and warms the atmosphere about 80 times more than carbon dioxide over 20 years. Additionally, in the atmosphere, it creates a positive feedback loop with another greenhouse gas, which is tropospheric ozone, which leads to more warming. About 60% of methane in the atmosphere is caused by human activity, which indicates a lot of potential for mitigation. Next slide. So some background, my project is a continuation of the California Methane Survey, which my faculty mentor participated in. It was the first systematic group of surveys to detect point source methane emitters in California. So a point source methane emitter is a feature generally less than 10 meters in diameter, and it emits concentrated methane plumes. The plumes were detected using the NASA Airborne Visible Infrared Imaging Spectrometer, next generation instrument, which uses absorption spectroscopy to detect methane from an aircraft. The survey utilized the Vista California dataset, which is a GIS dataset that was compiled using publicly available data on facilities that emit methane or that have potential to emit methane. Next slide. So some results from that survey were that 10% of the surveyed point sources were responsible for 60% of the detected emissions over every sector. So in my project, I'm focusing on the oil and gas sector, which attributes for 26% of the emissions found. So by documenting information on the emitting structures in the sector, we can reduce emissions. Next slide, please. So some problems we address in this project are that the data on what types of structures serve as point source emitters are not well known. What is also not well documented is where and what infrastructure are in oil fields. Additionally, the parent study found that emissions from the sector are about 60% higher than what is reported in national gas inventories. So the goals of this project are to locate and document the infrastructure for industry expert verification, the development of a systematic locating method to ensure all areas are checked, as well as developing a documentation method of the infrastructure that can be utilized by industry experts as well as future project participants. Next slide, please. So for the methodology, I use Google Earth Pro to locate infrastructure, and here's Kern County, which is the scope of my project. Next slide. An aqua grid layer was created to divide the oil fields up into equal sized squares, which have unique identification numbers assigned to each square, called the field identification number or the FID. Each FID is linked to the oil field in which it resides, and the oil fields are depicted using the purple boundaries. This allows for systematic division of the field in a trackable way. Next slide. As I would scan each grid for infrastructure, I place a numbered pin on my targeted infrastructure. The information linked to this pin are now the coordinates, the field identification number, and the oil field name. The diameter of the infrastructure can also be taken using the ruler function in Google Earth Pro. Next slide. So now that we have this information, how would you document it in a sortable, searchable, and usable way? Next slide. So for the documentation, all this information is extracted into Google Sheets that were designed for this, called the oil field tracker, where each section of information is sorted into a column and row. And this allows for the information to be sortable. And that's important to narrow down the focus of what you could be looking for, because the diameter especially could indicate what type of infrastructure something is. And the bottom legend shows different tabs that list out all the oil fields alphabetically, where you can indicate which ones were checked. And there are separate tabs based on the type of infrastructure that are being identified as well, where the pin information is being documented, and that's what we're looking at right now. And the cells are formatted to update to the legend on the right. Next slide, please. And that legend on the right will update to the summary tab of the whole tracker, which indicates what percentage of which oil fields were checked for which type of infrastructure. Next slide, please. And then here is just a sheet where participants in my position would track the oil fields we were assigned to check. Next slide, please. So now getting into some work that I contributed to this documentation process. I searched many oil fields for oil storage tanks, and I documented them on the oil tracker sheet. I also extracted data from other people's oil field tracker, and I also reviewed and verified data of others for secondhand confirmation. Next slide, please. I also identified oil pipelines in the Elk Hills oil field and mapped them using the drawing function in Google Earth. On the left-hand side is what one iteration of an above ground pipeline would look like, compared to the right-hand picture of what something underground would look like. Depicting these pipelines on the field serves as a guide to search for other potentially emitting infrastructure. So here is a blank picture of the Elk Hills oil field with just the grids on it. And trying to find infrastructure in here could be kind of difficult, as you'd have to look through each square. So next slide. And so if you add the pipeline layer, you can use the pipelines as a guide to infer surrounding structure instead of having to blindly look through each square. Next slide, please. And as you zoom in, you can see that where a lot of these pipelines have ended, there are a lot of infrastructure there. And this infrastructure is storage tanks. So this would be a good area to survey for emissions in the future. And this process is going to be helpful to find more areas like this while we're mapping. And next slide, please. I also searched for and documented separation units, which are pressurized chambers that separate liquid and gas well materials. In the previous survey, the structure was found to be one of the highest emitters in the sector. So the goal was that if we can identify these structures in all oil fields and determine if they are the highest emitters across all oil fields, targeted mitigation plans and policy can be developed centering this type of infrastructure. Next slide. So two of the oil fields that we targeted for documentation are Poso Creek and Kern River, which are very close together. The blue and green dots indicate wells, while the white circles indicate methane emissions. So a question we encountered was, why does Poso Creek have more emissions than Kern River, but significantly less oil wells? Some hypotheses could be that while Poso Creek is divided up into many small companies and Kern River is owned by one company, each company could possibly have an emission cap, so more companies means more emissions, or maybe smaller companies have more emissions exemptions. That's something to look for in the future. Next slide, please. I also aided my mentor in testing out new ways to document infrastructure for experts to later verify. We tried to put blank pins on every infrastructure we found and then export those pins to the experts, and they could identify each pin themselves without having to go through every field. But that wasn't very efficient for one person to have to preemptively do that in order for things to be identified. So instead, we decided that the experts would have to place the pins themselves and then upload a KML file to Google Sheets. So next slide. Some of the results of the work. So I made progress toward every project goal that we had and in terms of actually mapping the infrastructure and documenting them for verification. I also contributed to the development of the methodology by testing out new ideas and giving feedback. And this process will be used by future members of the project as well. There's also an infrastructure catalog being created currently for future project members to use. Next slide. Some future implications. First, I want to preface this by saying that the solid next steps in the process are to finish documenting the oil fields and then calculate what % of each infrastructure in those oil fields have emissions using data from the parent study as well. It'll also contribute to more targeted surveys in the future. The methodology can be implemented by future project members and hopefully other institutions and states. There can be direct mitigation with oil field operators. The diameter from tanks can be used in studies to see if that tank size has correlation with methane size, plume size frequency. And reducing methane emissions in the short term is important because these emissions can trigger an irreversible global warming feedback loop where the warming in the atmosphere melts Arctic permafrost, which in turn releases more methane, and that's a cycle. So next slide. There's some citations, and next slide, photo sources, next slide. I'd like to acknowledge my principal investigator, Dr. Francesca Hopkins, my advisor and mentor, Patrice Barnett, who I literally couldn't have done any of this without, Dr. James Sickman. This internship was possible due to the funding through his Fertile Futures grant, which is funded through the USDA, and Sebastian B. Rode, Frank Hershkowitz, Lawrence Kramer, John Landoa, and Ray D. Williams, who also helped document infrastructure, as well as California Energy Commission, which funded the project. And if there's any questions, I am here to answer them for 1 minute.
Thank you very much, Tiffany. That was a great presentation. If anybody happen to have any questions, you are welcome to type it into the chat, or you can raise your virtual hand and we can answer it, or actually Tiffany can answer it live. It looks like Dr. Hopkins has given you your seal of approval there. You do have one question. Why was Kern County selected?
You know what, that is a good question. I don't know the answer to that, but I can find out. But,
not a problem.
Yeah.
Well, and I think, yeah, we've actually run at our time here. Again, thank you very much. Oh, and Dr. Hopkins has answered your question. Most of California's oil and gas production does take place there. And one more question, which mapping software did you use?
So for the whole project, we used Google Earth Pro, and that's a free software anyone can download. And there's tools on there that you can use to place pins, make measurements, create polygons, create grids, and you can attach all kinds of links and data to whatever you create. So that was very helpful. GIS was considered, but this was kind of more convenient.
All right, thank you very much. Okay, we will be moving on. Congratulations again. And we will go to our next speaker of the morning, Ms. Martha Lopez. Thank you for logging in there. Your PowerPoint is up, so the floor is yours. Thank you.
Thank you. I just want to start off by saying that I live by the airport, so you might hear a few airplanes passing by. Hello, everyone. My name is Martha Lopez. I am an undergraduate transfer student majoring in biology here at UCR. I had the pleasure of working this summer in Dr. Daryl Jenneret's lab. My research project consisted of urban tree placement to reduce heat burden in disadvantaged neighborhoods. As some of you may know, trees have become an important component in the fight against climate change. Next slide. Thank you. Some of you may know trees have become an important component in the fight against climate change. The points listed on this slide here are a few qualities that have been identified to be positive results of urban tree placement. For the purpose of this project though, we narrowed it down to review an aspect that falls under the ecological component. Trees allow improvement within the communities and are a vital influence in tackling urban heat island. Urban heat island is recognized as an urban area or metropolitan area that is significantly warmer than its surrounding rural areas due to human activities, for example, the construction and presence of tall buildings. The temperature difference is usually larger at night than during the day. The severe climate change is one of the reasons why many cities have actually become interested in planting and maintaining trees in their neighborhoods, which is the reason why many organizations have been forming to address the situation. Next slide, please. Urban heat mitigation has been proven to correlate with high health risks as well as mortality rates in communities. The urban trees affect both air temperature and the radiant environment, so when trees are planted, they provide a cooling effect as well as shade. Although there are many uncertainties with how well the trees have been able to cool the environment, it is important the programs associated with the installation of the trees have an equitable, conscious effort to provide this option for all communities. The communities that have proven to show a larger portion of health risks like lung disease and cardiovascular have been predominantly in lower income or minority areas. The citizens in these areas have been exposed to heat at much higher rates than those in areas that have a higher percentage of tree canopy. Next slide, please. Thank you. So that brings us to the next portion of our study. What kind of questions could we form to understand how the tree planting programs were taking into account the amount of people that were suffering from health risks and what were they actually doing about it? We knew we wanted to develop questions that allowed the organizations to have concrete data-proven answers that open the conversation enough to discuss their strategies for disadvantaged neighborhoods in predominantly low tree canopy areas. So we produced these two questions. How are the organizations involved in urban tree planting and maintenance considering climate change in their practices? Specifically, how are low-income or minority neighborhoods being considered in treeing activities to mitigate the urban heat island? Next slide, please. Thank you. In order to have diverse and unbiased results, we needed to develop a plan that incorporated organizations in different regions that have distinct climate. In order to have various of options, the 3 cities that would take part in the study would be Los Angeles, California, Phoenix, Arizona, and Baltimore, Maryland. Within these cities, numerous of Zoom interviews were conducted, which included nonprofit organizations and city programs. Next slide, please. After conducting a well enough sum of interviews, it was important to be able to analyze and organize the recorded transcripts in order to clean up and comprehend the interviewees' responses. Since the interviews were recorded, it took some time to get through them and get them to be comprehensible without changing any dialect. Once the transcripts were finished, we went back and began to code by highlighting the passages we believed to be beneficial. We divided the open coding into categories or focus coding in order to have themes that will help pave the way to theory development where we are able to draw some conclusions regarding our research question. Next slide, please. In order to narrow down our results, we took responses from 3 programs, 1 from each city, and we found that although all 3 programs had become established in different environments, they shared a common objective. All organizations were motivated to provide tree canopy improvement in disadvantaged areas with lower tree canopy coverage and greater heat burden. Eric Dale from Tree Baltimore, for example, disclosed, if you overlay a map to show poverty levels, for example, average income there alone is a good correlation with where we are prioritizing and planting our trees. So we knew the programs were in fact considering the underprivileged neighborhoods, but how do they know where to plant the trees? Mr. Dill mentioned it in his response. They use online maps like the ones in this slide that reflect the areas that have low income neighborhoods based on the maps you see. You might say, well, I see a lot of green coverage, but the areas that have tree canopy are the darker green areas. The lighter shades of green are potential planting sites. Next slide, please. Beyond planting more trees, there was a specific emphasis on providing ongoing maintenance for trees planted in low-income neighborhoods. For example, Amy Esposito of Trees Matter in Phoenix discusses a program she is involved in, teaches the students how to plant the trees and care for them even though maintenance is the one that truly oversees them. The program still checks in and makes sure the trees are growing properly and are replaced if need be. When it comes to the community, Mrs. Esposito mentioned, we have an Ask an Arborist Facebook program because a lot of times a tree fails because someone doesn't have time to call an arborist or doesn't have the resources to fix a problem. And this gives people an opportunity to ask a question and troubleshoot quickly without spending funds. A common strategy that all the organizations shared was they try as much as possible to only plant trees that are low maintenance and require minimal tree knowledge. That way, it gives the tree a higher chance to survive and reduces replanting as well as prolongs tree canopy. Next slide, please. Future work would be needed to evaluate whether the treeing campaigns are finding success in the communities they are intending to serve, and if they are reducing the exposure of residents to excessive heat burdens. Ideally, if organizations are continuously allowed to plant and upkeep the trees, the tree canopy will increase for the communities and result in numerous benefits for the city. The temperature in the summertime would become cooler and bearable for children and seniors, as well as the air quality would improve, resulting in less health risk for the citizens. Trees have become an investment in the community. The trees have become an investment the community has to make in order to develop a healthier environment to live in. Next slide, please. I would like to take this time to fully acknowledge and express my gratitude to the incredible people who have allowed me to be part of this program and project. Thank you to the individuals from the program that helped organize such an inclusive and inviting experience. I would also like to thank the peer mentors who were always willing to go above and beyond to make us feel welcomed and like close friends. Thank you to Dr. Generette for allowing me to be part of his lab and offering insightful wisdom, and my colleague Dustin for being so kind and patient while providing great advice. I appreciate the National Science Foundation for the approval of the project that I work on. Finally, I would like to thank the people who made it possible for me to focus solely on this project and not finances. Mr. Kenneth and Nancy Gray, I am extremely grateful for your generous donation as well as your time. Thank you for everyone giving me all your attention to this presentation. And if you are able to, please plant a tree.
All right, thank you very much, Martha. I am absolutely inspired and thinking about what trees. In fact, if you look behind me, there's already a lemon tree that I did not plant. These are obviously from UCR, but yeah, if and when we do all step back onto campus, I think we'll have a better sense of appreciation for, yeah, a lot of the role or the big role that plants and trees do play, and especially with our university. And in fact, that's how we were founded, by a citrus experiment station. So does anybody, if you happen to have any questions for Martha, please feel free. You could raise your hand or type into this chat. We will give you a moment. While people are potentially typing up a question, so what was one of your favorite things about doing your research remotely for the summer, and what were your expectations and how were they met?
The hardest part for me was kind of getting my opinion out of it and actually being a scientist and not kind of forming my own opinions. Basically, I had to work with what I had, and that was a little bit difficult for me to not express my opinion.
Yeah, that's one thing. That's one thing I think we all have to work on, and that's a good thing with science as well. Is to an extent, when it comes down to it, it doesn't always matter about your opinion because it does come down to facts, and we're gonna, we're trying to find those facts. So Dr. Katie Burnett did ask, will you be able to continue this project? And I presume after the summer.
I actually don't know. I will have to ask Dr. Generette that.
Yeah, it's something to discuss.
Yeah.
I think Marissa here had a public service announcement that the city of Riverside gives a $25 credit towards a shade every March.
Oh, that's interesting.
Yeah, hopefully they continue that. In fact, I believe I did take care of that, or I did take advantage of that when I lived in Riverside, and it actually came as part of the utilities bill, where they just had an announcement. Yeah, and you just go to a few designated nurseries around the city, and they will either give you, they'll give you credit toward a shade tree of your choice. I think Marissa's a huge citrus fan. So anybody there happens to have any citrus trees, she will take any and all of them. I think I gave her one of mine, and yeah. Oh, and same thing with Dr. Katie Burnett. They bought several trees using the credit. All right. Well, thank you very much, Martha.
Thank
great
you.
Presentation. Glad to have you on board, and we will see you around.
Bye.
Okay. So our next presenter is going to be Ms. Jalin Tunel. Thank you for turning on your camera. Oh, I like your background.
We're matching. We're twins.
All right. Well, your PowerPoint is up and running, so the entire floor is yours. Thank
awesome. You. Good morning, everyone. My name is Jalin Pinello, and this beauty you're seeing on the screen is Pieris rapae. So today we'll be talking about how this butterfly took over the world and the effect that it had on its own genetics and physical appearance. So first, we're going to go over what Pieris rapae is. Then we are going to start discussing a previous study done that really inspired what we're doing. And lastly, we'll cover my research and what I accomplished during RISE Summer Bridge to Research program. But before we dive into the deep end, let's go over some background on the next slide. So Pieris rapae, or as you might recognize it, the white cabbage butterfly, is an extremely invasive butterfly and is found on every continent except South America and Antarctica. It's rather inconspicuous, and it took a while for scientists to notice just how detrimental this butterfly can be on cruciferous crops. And by cruciferous, I mean leafy greens like cabbage, bok choy, Brussels, cauliflower, and broccoli. So let's move on to the next slide. So let's look at our past research. In 2019, a paper covering a huge collaboration between ordinary citizens and scientists was published. This paper birthed the Pieris Project, an international citizen science project made up of over 150 citizen participants. And you might be wondering, what use can normal citizens have with scientific research? The answer is a lot. Ordinary people are so important and contribute so much to science, in other people's research and in my own. In this paper, the specimens gathered from these recipients accounted for a huge chunk of data collected and analyzed. From this data, scientists were able to map out the origin of Pieris rapae and their entire invasion history. So let's bring our attention to the map on the screen. From their data, they were able to determine that the mother population was Europe, identified by the dark green dots in the middle. There were two initial divergences from this parent population. One spread to Asia, identified by the orange dots, then up to Siberia. Those are the brown dots. And then the second divergence spread to Eastern North America. Then from the East Coast, it spread to the West. From the West, they spread to Hawaii and New Zealand, and lastly, New Zealand spread to Australia. These butterflies were the ultimate hitchhikers, using human trade routes like the Silk Road to catch a ride from place to place. Along with using the specimens to map the invasion history, scientists then used them to perform genetic analysis. Let's look at the next slide. Using mitochondrial DNA, which is present in every single organism on Earth, they found over 22,000 single nucleotide polymorphisms, or a SNP. Each SNP represents a difference in a single DNA building block called a nucleotide. So for example, a SNP may replace the nucleotide adenine with the nucleotide thymine in a certain stretch of DNA. They also found that each of the 7 regions had their own genetic cluster. So that means these butterflies in these 7 regions are more related to each other than the parent population. So during their genetic analysis, they also observed that the butterflies underwent an intense bottleneck, which, as you can see from the picture at the bottom of the screen, the genetic diversity bottlenecked whenever the population founded a new place. And these genetic findings led us to our research question. Let's look at the next slide. In our research, we asked the question, are there any physical differences between the California and New Zealand populations of Pieris rapae? We're investigating this question because the West Coast, specifically San Francisco population, is the mother population to New Zealand. And as you can see from the phylogenetic tree on the right, the baby pink North America branch turns into the fuchsia colored New Zealand branch. These two populations are genetically different from each other. And we want to know if these genetic differences led to physical or phenotypic differences as well. So let's look at another slide. How would we find this out? By collecting data. As we researched these butterflies, we found that they were sexually dimorphic. So the different sexes looked different. The males generally had 1 spot, as you can see from the top butterfly, and the bottom is a female. It has 2 spots. Beyond this, we collected measurement data from the wingspan of both populations to compare later. We did all our measurements using a program called ImageJ, which you can see at the bottom of the slide, and I analyzed over 1,200 images and measured wing length, body length, and antenna length for further use. So let's talk about where we acquired this data. We used two databases for this project, the first being idigbio.org. idigbio is an online database of digitized museum samples from around the world. We expected there to be a lot of photos. So we set a criteria. That criteria being it needed to have a map point and obviously it needed to have a photo. From California, there were there were exactly 204 specimens, but only 53 were useful. And in New Zealand, there were no photos digitized from museums at all. Let's look at the next database. The next database we used was photos from the app Inaturalist. This is on the site GBIF. So Inaturalist is an app where people take photos of species for identification. From this database, we found 826 occurrences of Pieris rapae in New Zealand and over 3,000 occurrences in California. But just because these pictures exist doesn't mean they're usable. Let's see what I mean on the next slide. Not all pictures are created equal. So if you look at the top two, those two, I can't see the body, I can't see the antenna, and I can't see the spots. And the bottom two are not even butterflies yet. So when I analyzed those 1,200 from New Zealand, I only got 31 useful ones. And for California, I decided to just match this number. Once we collected this data, we analyzed and compared it. Let's look at the next slide. Now look at these beautiful graphs. These graphs were created with all wing measurements relative to body size. So let's bring our attention to these graphs 1 and 2. They show individual populations, comparisons of females and males. In both data sets, the data mostly overlaps, but if you look at the orange dots on both graphs, the female butterflies show more wing size variation, while the males stay within a certain size range. The females can be both much smaller and much larger than the males. Let's look at the next slide. Graph 3. This graph shows a comparison of females from both populations. We did this because New Zealand butterflies looked like they're going to be larger than the California butterfly population, which were already larger than the California males. Were New Zealand female butterflies even bigger than the California females? The answer is yes. From our graph, we saw that New Zealand females were much larger than even the California females. And if you look at graph 4, this graph shows all butterflies, male and female, from both populations, the orange being New Zealand and the blue being California. From this graph, we concluded that while the data overlaps most of the time, the butterflies in New Zealand were much larger than the butterflies in California. Let's look at the next slide. After analyzing the butterflies, we found out that New Zealand females got much larger than all other butterflies from both populations, male and female. Let's go to the next slide. So our conclusions from our data sets were, we found that butterflies in New Zealand can grow larger than California butterflies. We also found that there is more variation in wing size in female butterflies in both populations. And I personally concluded through this project that citizen science is extremely important. Without the help of normal people, we would not be able to do this project. And remember that anyone can contribute to science, and you should download iNaturalist and go take some pictures of some animals and plants. Let's go to the next slide. As this presentation comes to a close, I'd like to discuss what our future directions would be if we were able to continue our research. We would compare our findings to the European source population and investigate the reason for the size difference between the New Zealand and California. So we want to look at factors such as diet, climate change, and the environment overall. Let's go to the last slide. Here's my works cited, and then actually one more. All right, these are the people that I'd love to acknowledge, and they're the only ones that made this possible. So again, my name is Jalen Pinello, and thank you so much for tuning in. I'll be taking any questions.
Well, thank you very much. That was an exciting presentation, a lot of great energy, and you got Dr. Burnett's seal of approval.
Oh, thanks.
Right away. I'm sure she is extremely proud. Does anybody have any questions for Jalen here? Please go ahead and type in the chat. or raise your hand, your virtual hand, and we can have you answer live. Sounds like you had a lot of fun.
Yeah, I love presenting, actually. Okay.
So you do have one question from Radhika. Does the proliferation of the cabbage butterflies also affect certain animal species?
I'm sure there's a lot more food to eat because there are a lot of insectivores probably eat them, but we actually didn't really look into the effect that they have on other animal species, and that could be something we do in the future if we were to continue.
All right. Tiffany has asked, do you have any advice on how to take usable pictures on Inaturalist?
It depends what you're taking a picture of. We had some really awful pictures. Make sure it's not blurry. Make sure they're not in motion, and make sure that that it's as identifiable as possible. Those would be my main things.
Yeah, okay. And a couple short questions here. Marissa's just asking, who happens to identify the species?
It's like,
on iNaturalist?
Yeah, it's like a crowd identification of hobbyist scientists who happen to have the app. So think of like Reddit, how it's a whole bunch of people talking at once about something and there's a consensus. It's kind of like that, but there are a lot of scientists that actually use the app, and then hobbyists as well.
Okay. And lastly, Jay is asking, is there a website for citizen science?
I don't think there's a website specifically. You might be able to find some apps. And if you were interested in the citizen science aspect of the Pieris project in particular, you can go to thepierisproject.com.
All right, awesome. Thank you very much, Jalen. Great presentation.
Thank you.
We're going to go on to our next presenter, Mr. Ethan. And if you could turn on your camera and you get a second. Thank you very much, sir. So your PowerPoint is up and running. The floor is yours. Please take it away.
All right. Thank you very much, Mr. Tran. Good morning, ladies and gentlemen. I'm thrilled to be joined with all of you here this morning. And though it will not be the same online as it is in the auditorium, I am incredibly excited to discuss the remarkable work conducted over these past 8 weeks, specifically as it pertains to the patterns of evolution of the GATA transcription factor family in nematodes. Next slide, please. For a quick overview of nematodes, they are commonly known as roundworms, and there are millions of species estimated in this clade. It includes the model system Caenorhabditis elegans. Therefore, as it is a model system, it is studied with the belief that it will provide a greater understanding of biological functions in other species. Next slide, please. The image here depicts the 5 main clades within the Nematoda phylum. The closer 2 species are on the tree, the more closely related they are, while the further apart they are, the more unrelated they are. For example, species in clade 4 are more related to species in clade 5 than they are to species in clade 1. Caenorhabditis elegans, as with the majority of species in this study, though not all, is in the top clade called clade 5. Next slide, please. For some background on God effectors, they are a specific type of transcription factor, and transcription factors control the rate of transcription, which is the process of converting DNA to RNA. Transcription factors can accomplish this by either promoting or inhibiting the binding of RNA polymerase, the enzyme responsible for converting DNA to RNA. Through its control of transcription, transcription factors can control gene expression, whose objective is to change a gene into something useful, such as a protein. God effectors in particular bind to the sequence GATA and primarily regulate cell and tissue identities in animals. The section labeled A in the figure on the right of the screen is of a zinc finger, which appears like a loop, and everything to the right of this loop is the basic domain. The single letters forming the loop are amino acids, and if you look, there is a highlighted ZN with 4 lines connecting it to 4 Cs. The ZN stands for zinc, which is where the structure derives its name. Section B is of a GATA DNA sequence, in which each letter represents a nucleotide. And as you can see, the letters G, A, T, and A are highlighted. Next slide, please. The objective is to better understand the evolution of God effectors in nematodes. Next slide, please. This project was conducted using a wide variety of software, some web-based and others run locally on the computer. cnarbitis.org, parasite.wormbase.org, and wormbase.org were all used to retrieve the sequences of the various species. FASTA files, a file to compile the sequences retrieved from the previously mentioned websites, are required to run the analysis of the sequences. Numerous Python scripts were written to aid in the construction of these FASTA files. These FASTA files were then used in the basic local alignment search tool, commonly known as BLAST, to locate the best matches of God effectors in the various species. Specifically, we use BLAST-P, which is a protein-to-protein search, and T-BLAST-M, which is a protein-to-nucleotide search. Both the public web-based NCBI BLAST server and the command line version of BLAST were used. Python scripts were again used to compile the results from the numerous BLAST searches into one coherent FASTA file. This FASTA file was then aligned by the multiple sequence comparison by log expectation, commonly known as muscle alignments in the MegaX program. Finally, phylogenetic trees were created by both the MegaX program and RAXML. Next slide, please. Using the C4 zinc fingers and basic domains, we were able to perform muscle alignments and utilize RAXML to construct maximum likelihood trees to deduce patterns of inheritance amongst the different species and clades. Of the 5 previously mentioned clades, clade 2 was not sampled as the sequences were not available. Next slide, please. On this slide, we have some examples of phylogenetic trees created by both RAXML and MegaX, and an alignment created by MegaX. The main image on the left is of a tree and is made in RAXML using the specific God effector called ELK2 in a variety of species. As previously mentioned, the closer 2 species or clades on the tree are to each other, the more closely related they are. For example, C. brens and C. trops L2s are incredibly similar, while C. vires L2 is much less similar to these 2 species. The other phylogenetic tree on the top right is made in MegaX, which differs in its algorithm for constructing trees compared to RAXML. The tree was made using different goddess from different species to determine the relatedness between the different types of goddess. The alignment at the bottom right is also made in MegaX. The top row shows the consensus sequence of amino acids. And all amino acids, which are conserved amongst all species, are labeled in the yellow highlight. Where differences occur, they are noted with the substituted single letter amino acid, while amino acids that match the consensus are labeled with a dot. Next slide, please. The tree now shows the number of God effectors per clade, bar clade 2, in the previously shown phylogenetic tree. There is an increase in God effectors as you move up the clades, as clade 1 has 2 to 3 God effectors, and clade 5 has 4 to 5 God effectors. Next slide, please. This phylogenetic tree shows the 3 classes of God effectors. In this phylogenetic tree, rather than species grouping together, we have the various protein sequences grouping together. In blue, we have the 2 zinc finger structure. In red, we have the 1 degenerate and 1 zinc finger structure. And in yellow, we have the 1 zinc finger structure. The phylogenetic tree shows clear and distinct clusters of each goddess structure. Take the giant blue rectangle, for example. All of those are all 2 zinc finger structures. Each type of goddess factor structure clusters with the same type of goddess structure found in other species, suggesting that these structures have evolved from a common ancestor. Next slide, please. The driving force behind the creation of the previous phylogenetic tree are the similarities in the conserved amino acids amongst the various species. The amino acid sequences of the 3 structures of goddess zinc fingers are shown in the image. The red bars label the basic domains, and the blue bars label all the proper zinc fingers, while the green bar labels the degenerate finger. As previously mentioned, all amino acids which are conserved amongst all species are labeled in the yellow highlight. As seen by the large amount of yellow highlight, the 2 zinc finger structure, located at the top, has the strongest conservation of amino acids. In the middle structure, the degenerate and 1 zinc finger structure, the degenerate zinc finger has virtually no conservation other than the 4 cysteines, while the proper zinc finger has been well conserved. The bottom structure, a single zinc finger structure, has very little conservation when compared to the other two structures of zinc fingers. The degenerate zinc finger likely does not bind DNA, while all other zinc fingers do. Therefore, amongst all the DNA binding zinc fingers, the single zinc finger structure is the least conserved. Next slide, please. For conclusions, we have identified three distinct classes of Gada structures. As previously mentioned, they are the two zinc finger structure, the one zinc finger and one degenerate zinc finger structure, and the single zinc finger structure. We have also concluded that there was likely one of each type of zinc finger structure at the base of the phylum. Virtually all duplication is in the single zinc finger structure. The other two structures are rarely duplicated. A correlation between the number of God effectors and developmental time may exist, as over 10 God effectors have been observed in Synarditis elegans, and the species develops rapidly. It may be that as organisms develop faster, a benefit to more precise control of gene expression may be beneficial. Next slide, please. I would like to acknowledge Dr. Morris Maduro for his assistance throughout the entirety of this project, for both his expertise and encouragement. I would also like to express gratitude to the RISE staff for the phenomenal work that they have done to guarantee that the program, although online, provides for an enriching experience. Finally, I would like to thank the CNAS alumni and RISE donors, specifically Mr. Kenneth Gray and Ms. Nancy Gray, for the funding that helps to make events like today's possible. Next slide, please. Thank you very much for sharing your time with me today, and I hope you enjoy the other fantastic presentations throughout the week. I open the floor to any questions. Thank you.
Thank you very much, Ethan. Well, I think we all enjoyed your presentation very much, and you already have one question coming in from Ms. Fatima. Yeah, she would like to know, what did you enjoy most about this experiment?
Yeah, thank you, Fatima, for your really great question. I think it's really important because, it is summer, and I think, during summer, you want to have a little bit of fun. I think the most enjoyable part of this experiment would have to be, a greater understanding of how species develop over time, because it really is interesting to understand how over time species change, how they evolve to suit new environments, and how their genes change over time to reflect these changes.
All right. Thank you very much, Ethan. Again, great presentation, and we will move on to our last presenter for this session before the break, Mr. Kendrick Davis. If you have a chance, please turn on your video. Thank you very much. All right, well, your PowerPoint is on the screen, so Sweet. You can start.
Can you hear me all right?
Your leisure. We hear you perfectly. Thank you.
Okay, awesome. Thank you. All right, so good morning. My name is Kendrick Davis, and I will be going over understanding the biochemical and mechanotransduction signals that regulate the sonic hedgehog and hippo pathways. And I worked in Dr. Morgan's lab, and also for the presentation, I will focus on the mechanisms and existing mathematical models that describe the dynamics of both of these pathways. Next slide, please. Thank you. So both the hippo and the hedgehog pathways were first discovered in invertebrates, specifically the melanogaster Drosophila fruit fly. For the hippo pathway in vertebrates, the homolog MST12 was later discovered, and transcriptional regulation of YAPTAZ, which is later downstream, those genes regulate organ growth and size. And for the sonic hedgehog pathway, that activity and concentration, the concentration and regulation of that activity leads to homeostasis of cells and tissues in the nervous system and patterning of limbs. For the hedgehog pathways, the three overall, Indian hedgehog, desert hedgehog, and sonic hedgehog, each of them have been shown to play roles in maintaining stem cells throughout the body. Next slide, please. Now, for the hedgehog pathway mechanism, I decided to focus on the off state first. here in the picture, figure A on the right, you can see patched and smoothen. Those are both transmembrane proteins. Patched is a 12-pass transmembrane protein, and I believe smoothen is 6. In the off state, patch 1 inhibits smoothen. Because of this stationary phase, KIF7 kinase family member 7 stays at the basal body of the primary cilia, which you see in the middle. KIF7 stays at the basal body, and later in the on state, it actually moves to the tip of the primary cilia. Now, on the other side of the primary cilia, the right side, you can see GPR161, another transmembrane protein. That presence on the membrane shows or increases CAMP activity. CAMP is a cyclic adenosine monophosphate, which helps with phosphorylation of the pKa cascade. pKa, CK1, and GSK3 beta are kinases which help with phosphorylating Gli2-3. And once Gli2-3 is phosphorylated, it detaches from SUFU. Once it has detached from SUFU, a proteasome comes in and excises or cuts the Gli2-3 phosphorylated into its repressive form, which is Gli2-3R, and that cannot undergo transcription, which is an overview of the off state. Next slide, please. For the on state, as you can see, Hedgehog attaches to patch 1, and patch 1 moves away from the basal body on the plasma membrane of the cell. And then because of this movement, smoothen is allowed to be activated, which is activated by DLG5, and DLG5 helps with translocating it. EVC and EVC2 also help with further movement along the primary cilia up to the tip. And then because of this movement, KIF7 can localize to the tip of the primary cilia, bringing the SUFU and Gli2-3 complex along with it. As you can see on the right, well, on the last slide, GPR161 was attached to the plasma membrane near the basal body. Now it is moved and detached from the plasma membrane into the cytoplasm. Both patched and GPR161 are recycled by a proteasome in the cytoplasm. Because of this and GPR161 not being near the basal body, CAMP activity decreases, which inhibits the pKa kinase cascade from phosphorylating Gli2-3. Gli2-3 then naturally disassociates from SUFU, and it becomes its active form Gli2-3A, and then that leads to transcription of the sonic hedgehog genes. Next slide, please. Now for the Hippo pathway, this is regulated through, well, this is based off of a paper that hypothesized that this is controlled by the extracellular matrix and adherent junctions and forced transmission through those proteins on the perimeter of the cell, the plasma membrane. Adherent junctions are made of E-cadherin, alpha-catenin, beta-catenin, and a cluster of other proteins for structural integrity. FAC is an adhesion kinase molecule which begins that cascade once a certain force, like 1 kilopascal, or a certain force comes in and is transmitted between cells. FAC is released, which helps with the phosphorylation cascade. That ends up phosphorylating row A, which you can see in the middle, and then row attaches to MDA or ROC. MDA is a formin which helps with polymerization or nucleation of G-actin into F-actin, and F-actin helps with transcription of the YAPTAZ genes. And then on the other side, row A attaching to ROC, it helps with phosphorylation of light chain myosin into myosin, and then that helps with activation of the YAPTAZ genes as well. And then the third mechanical portion is the main Hippo pathway, which is driven by Merlin, which is a cytoskeletal protein that helps with the kinase cascade, and MST1, and then LATS phosphorylation, and then LATS actually inhibits YAPTAZ transcription by phosphorylating YAP outside of the nucleus. Next slide, please. That's a rough overview. And here are the materials and methods for what I had done. So I used MATLAB's ODE solver, ODE45, to compute the 10 ordinary differential equations that were describing the sonic hedgehog pathway. And in that paper, they had patch, inhibitors of patch, I believe NKX 2.2, then GLIFL, GLI23A, and GLI23R in their active and inactive states. That's what the equations describe. And then graphs of those concentrations over time were recreated from that paper, the Cohen paper. Next slide, please. Here is a figure of all 10 of the equations. They're driven by, oh, my apologies. This is the only figure in the paper that was actually available, so it's very blurry and very small, or slightly blurry and pretty small. And this was in the supplementary section, but yeah, each of these equations are described by transcription rates, degradation rates, translocation rates, activation rates, and conversion rates between different proteins, describing them. Thank you. Here are my resulting graphs. They are similar, but off by certain factors, but we're working towards improving them and their concentrations that they're describing. Next slide, please. And for the ongoing work, I will be incorporating another mechanical model that describes, rather, it quantifies the adherent junctions that play a role in the Hippo pathway based off of the cell size. And yeah, the cytoskeletal components are quantified based off of the cell size and its geometry. Further screening of those parameters that we used for the sonic hedgehog pathway will be done in sensitivity analysis using probability statistics, possibly approximate Bayesian computation for that sensitivity analysis will be done. My professor sent a paper on that, and then coupling of those pathways to further understand some of the key proteins involved that drive cancer growth and proliferation, and also just the cell's activity overall will be done. That's the goal with the two pathways. And then here are my references. Thank you. And my acknowledgements. I would like to thank Dr. Joshua Morgan for his guidance through this process. It's been tough but fun, and creating this project and moving toward the ongoing work for greater progress this year. I would like to also thank Nitran and Dr. Brown and Dr. Richard Cadrullo. Also, Dr. Connie Nugent for the resources and the lasting effect that this research program has provided me with the information and my future endeavors regarding graduate school. And last but not least, I'd like to thank the CAMP program and the National Science Foundation for this opportunity and funding. Thank you.
Awesome. Thank you very much, Kendrick. Great presentation and right on time. Looks like you got another seal of approval here from Jasmine. Does anybody happen to have any questions for Kendrick? Please feel free, type it into the chat here or raise your virtual hand. Love to hear somebody else besides myself. But of course, we could hear from Kendrick all day, and actually the presenters. This morning's presenters you guys were all amazing. That was great, very engaging, and a great way to start off the program. Yeah,
thank you. I got a month start ahead of this. Oh, to answer Dr. Brown's question, my favorite part was, I did like the CAMP part, the cyclic adenosine monophosphate, and that camp. Pretty funny. But also my favorite part was the fruit fly. I didn't expect that, and it's used in a lot of lab studies, which is pretty cool. I found that really interesting. And also the differential equations are fun. I like the math.
All right. And I posted Dr. Brown's question in case anybody was wondering what Kendrick was responding to.
Oh, yeah.
Okay. No worries. All right, Dr. Nugent's jumping in. Well, time has come for our very first break of the morning session. So we will start off with 5 minutes to go run to the bathroom. For me, I'm grabbing 8 more cups of coffee, and just stand up, take a stretch. We will reconvene in exactly 5 minutes, and we'll start our next session of both oral and poster presentations. So thank you very much for tuning in. We'll see you in a little bit. Thank you, Kendrick. All right. Welcome back. I don't know about everyone else, but that truly felt like the shortest 5 minutes I've ever had. So we have our next presenter up and running who also spoke this morning. So Mr. Alex Rojas, your presentation is now on the screen. Thank you for being prompt. And I will give the floor to you, so take it away.
Thank you, Nee. Hello again. Good morning. I hope everyone's doing well and staying safe again. My name is Alex Rojas, and this is my presentation on changes in California air pollution relating to the COVID-19 global pandemic. Next slide, please. So, what causes air pollution? Well, humans definitely account for a large amount of emissions, but air pollution can also be caused by or spread through natural events. Next slide, please. A few natural sources can include through volcanic eruptions, wildfires, thunderstorms, and winds, to name a few. While there may not be much that we can do to prevent these natural sources from generating pollutants, we can decrease the amount of anthropogenic emissions. Next slide, please. Photochemistry allows reactions to occur in the atmosphere between molecules such as ozone, nitrogen oxides, volatile organic compounds, and other chemicals in the presence of UV light or sunlight. In addition, warmer air and intense sunlight, as seen in summer months, allow these atmospheric reactions to occur much more frequently. The following reactions show how nitrogen dioxide can be a precursor to ozone production and vice versa. I'll go more in depth on these reactions in just a little bit. Next slide, please. These are the 4 standard or criteria air pollutants you see here are they'll be discussed in the presentation. Ozone can be helpful in the stratosphere as it protects us from the sun's harmful UV rays, but ground level ozone, found here in the troposphere, is known as smog and can be harmful towards respiratory health. For carbon monoxide, exposure in low levels can cause problems such as headaches, dizziness, or even chest pain in individuals with heart disease. While exposure in high amounts can be fatal, it is unlikely to experience a high enough concentration through the outdoor air alone. Nitrogen dioxide is primarily produced from cars, trucks, power plants, basically anything that combusts fossil fuels. It can also further aggravate respiratory problems, leading to excessive coughing, wheezing, or shortness of breath. Fine particulate matter consists of matter that is less than 2.5 micrometers in diameter. For size comparison, that's at least 30 times smaller than the diameter of a single strand of hair on your head. These pollutants can easily be inhaled and penetrate the lungs, damaging their function. Next slide, please. The COVID-19 pandemic, as I'm sure we're all well aware of, has required millions of Americans to stay in their homes. Non-essential businesses and factories were forced to close temporarily, and people have had to follow guidelines like social distancing to prevent further spread of the virus, just like we're doing right now with this remote Zoom webinar. This study focuses on the effect that this great reduction in human activity has on the air quality in California when compared to previous years. Next slide, please. For the methods of this study, all data was compiled from the California Air Resources Board. Each location used several monitoring sites to relate an average concentration of air pollutants for comparison. The data was primarily collected from ground level sites throughout California to generate a more accurate estimate of the air quality that we actually breathe. Next slide, please. The map on the left shows the counties and locations that were studied. So you have Riverside, Los Angeles, Kern, Fresno, and the San Francisco Bay Area. For the sake of time, I'll be mainly going over the daily average results of Riverside County. To help with data comparison, the monitoring sites were also sorted into either a high traffic or low traffic area. For the results indicate that the concentrations of nitrogen dioxide and fine particulate matter decreased the most during the pandemic from mid-March to May. To be specific, March 19th to May 8th. After around early May, these levels of these same pollutants began rising again and even overlapping with the concentrations recorded from 2018 and 2019. Next slide, please. So figure 1 shows the daily average of ozone levels for Riverside County in both areas of low traffic on the left and high traffic on the right. Note the levels between the dates of March 19th and May 8th. These dates are during quarantine, but I'll discuss shortly why these specific dates are important. Now, while these levels do seem lower than previous years, it is not nearly as evident as you'll see with some of the other pollutants. With figure 2, carbon monoxide levels, these levels show a little more of a clear decline between those quarantine dates at times, though they do fluctuate. Now for the next graph, figure 3, it shows a well-defined decrease trend of nitrogen dioxide between these dates when comparing it to levels from 2018 to 2019. And this pattern was evident in all of the locations that were studied. Similarly, the following figure, figure 4, shows this clear decrease trend with the concentration of PM 2.5 for this year. While the data for Riverside County's low traffic sites at the beginning of the year, so the bottom left, was not available, I still had plenty of data from the other counties and locations that supported similar findings. These tables display the average percentage difference of the data from 2020 versus 2018 to 19. This is based on a daily average of all the data from all 5 locations throughout California. The table on the left shows very little change in emissions before the pandemic, so January 1st to March 18th, with only a 1% and even 12% change, while the table on the right shows more substantial changes during the pandemic. Specifically, NO2 and PM 2.5 show a significant percentage decrease with negative 23% and negative 19%, indicating that the emissions of these pollutants during the quarantine period have been consistently lower than previous years. Next slide, please. Now here's where those important dates come back, March 19th and May 8th. March 19th was when the stay-at-home orders went into effect for the state of California, and shortly after, the ambient levels of NO2 and PM 2.5 showed a decrease when compared to previous years. On May 8th, those same orders were modified, allowing restrictions to be lifted and businesses to start reopening. Around this time, the levels of NO2 and PM 2.5 began rising again, even matching with or exceeding the levels from 2018 to 19. These findings support evidence of a direct relationship between the concentration of these pollutants and anthropogenic activity. Next slide, please. Here's the mechanism for those photochemical reactions earlier. They show an inverse relationship between ozone and nitrogen dioxide. So as you can see, there's NO2 all the way on the left and O3 all the way on the right. Now, as you follow the arrows, the NO2 molecules can react in the presence of sunlight or UV light to separate and combine with other molecules in the atmosphere, like oxygen, the O2 molecule on the top right, thus creating ozone. Similarly, these O3 molecules can combine and react with other atmospheric molecules, eventually creating nitrogen dioxide. As concentrations of NO2 rise, more ozone may be produced in the presence of sunlight, and as the levels of O3 rise higher, more NO2 molecules may be produced to balance out these reactions. So this cycle of atmospheric reactions does depend on a multitude of factors, like weather and the time of day. So afternoons typically have a higher ozone concentration than nitrogen dioxide. Next slide, please. During the quarantine months, the data indicates most pollutants like NO2, CO, and PM 2.5 express lower concentrations. O3 levels, on the other hand, had very little change. This is due to that inverse relationship between NO2 and O3, further supporting those atmospheric reactions. With all this, again, the results show the response towards the COVID-19 pandemic did cause a decrease in air pollution throughout California. This study helps to provide insight on what it may take to decrease air pollution, as well as identifying and understanding what specific activities and processes should be changed in order to improve the well-being of all life on this planet as we take the next steps to create a cleaner future. Next slide, please. In conclusion, further research may narrow down specific sources of harmful emissions so that we may make these necessary changes to better our planet and decrease future emissions. Next slide, please. I'd like to thank my faculty mentors, the RISE program, and coordinators, as well as all of the generous donors. Thank you again, and I will open up the Zoom meeting now to any questions.
Thank you very much. Great job, Alex. If anybody has any questions, please feel free to type into the webinar chat or raise your virtual hands. And I think comment from Tiffany there, and some people may still be typing. what was probably some of your challenges of jumping in to this research, since this is your first experience as an incoming transfer over the summer?
Definitely, I think it would be trying to figure out how to interpret all of the data into neat, clear graphs, because it was just so much data, and a lot of the data ended up looking really noisy. So that's why I was able to find out how to make a nice trend line that just shows the average amounts in a much more clear manner.
All right. And Dr. Zhang has given his seal of approval with a comment to you as a panelist. All right. Well, if there are no additional questions, again, thank you very much. Great job, Alex. We'll be moving on to our next presenter. Okay. So our next presentation is actually a group, all incoming transfers that were assigned for one specific lab. So we will have Ms. Nicole Shad, Mr. Jesus Kamba, and Ms. Alexandria Richardson by committee. So I will let you guys have the floor.
Thank you, Nee. Good morning, everyone. Before we begin into today's topic, I would like to ask, do we have any guesses what our topic might be?
Making a line.
Making a line. Well, let's find out in the next slide. Unfortunately, it is not making a line, but rather a very interesting topic. We did statistical data analysis for COVID-19. The research was pioneered by Professor Wei-Sin Yao, and it was done under Alexander Richardson, Jesus Kamba, and myself, Nicole Shad. Next slide, please. For some background, the novel coronavirus, as we all know, COVID-19, stems from a known coronavirus. And the first COVID-19 case was in Wuhan, China in late December 2019. Since then, CDC has done a lot of research and has found that older adults, 65 plus, minority groups, and males are at a higher risk of dying from virus-related infections. Now, why are we interested in this topic? Well, approximately 1 million people have died globally, so this is an issue that needs to be addressed. Next slide, please. According to CDC, 8 out of 10 COVID-19 deaths reported in the US were of people that are between 65 and older. Next slide, please.
Similarly with gender, more men than women have died of COVID-19 in 41 out of 47 countries, representing 2.4 times higher among men than women.
Across the United States, the majority of cases are made up of Hispanic people, American Indians, Alaskan Natives, and African Americans. The majority of deaths have been reported as being African American.
The purpose of our study was to formally test whether CDC claims hold true in local areas such as California and Texas. The objective was to find factors and covariates and study whether or not they had significant impact on daily COVID-19 cases and deaths. And the data was used to build regression models to predict daily cases and deaths.
The methods used to analyze this data was through our software, and we did tests of the daily cases and deaths. The following tests are the Friedman test, the Wilcox test, and the Quaid test.
The data was standardized by taking the ratio of cases and deaths in relation to the percentage that the group made up in California. Using this data, we created categorical values for ethnicity, gender, and age, which was used to form polynomial and linear regression models to predict the numbers of new daily cases and deaths. Next slide. These graphs show the daily new cases and deaths in California over a period of time going from April to mid-August this year. From this graph, you can see that the majority of new cases are made up of the age groups 18 to 49 and 50 to 64, while deaths are made up of 65 and older. These next graphs show the daily new cases and deaths in relation to gender. As you can see from the top graph, there's minimal difference between the numbers of new cases between male and female, while for deaths, there are slightly more male deaths than female. Lastly, this graph represents the numbers of new cases and deaths based on differing ethnicities. There are majority Latinos and Native Hawaiian and other Pacific Islanders that are infected by COVID, while there is no apparent trend in ethnicity for California in relation to deaths.
Next, we formally tested for cumulative differences between ages and gender to look at positive cases and deaths. As you can see in the graph above, we have that the people that were between 18 to 49 had the highest number of daily positive cases, while for deaths, the trend shows that 50 to 64 had the highest number of deaths, with 0 to 17 age group being almost negligible. Next slide, please. As we can see here, for daily positive cases from April 22nd to August 17, we have that females have had more positive cases than males, whereas for daily deaths, males have died at a higher rate than females. Next slide, please. Which comes to the conclusion that we did a p-value test and we found that the p-values were less than 0.05. And so we did a post-hoc analysis as shown in the table on the slide. We saw that pairwise comparison showed us a p-value of less than 0.05, which means that we had significant differences amongst these factors.
Similarly with age and gender, we also studied the different ethnic groups, both the infectious rate and the mortality rate, to find if there was a difference among one or more of the groups. And as seen in the results, the p-value that we obtained is a lot less than 0.05, meaning there's a group among one of the ethnicities. Can you go to the next slide, please? And therefore, we did a post hoc Friedman test to find which of one of these groups was different. And we actually found out that all of the groups, all of the ethnicities, both in infectious cases and then mortality cases, had a p-value of less than 0.05, and the letters represent the ethnicities. Can you go to the next slide, please? And this is the p-values for the mortality cases for all of the ethnicities when compared to one group to another. So therefore, we have a p-value of less than 0.05, leading us to reject the null hypothesis and conclude that all three factors have a significant impact on the local COVID-19 cases and deaths.
So pictured here is an example of one of our polynomial regression models comparing daily new cases to gender in California. Looking at the highlighted p-value for gender being 0.624, we can surmise that gender is not a significant factor that affects the numbers of daily new COVID cases. When creating these models, we took note of p-values looking for values less than 0.05 to determine statistical significance and R-squared for best fit. So the other state that we looked at was Texas. These pie charts represent the percentage makeup of COVID-19 cases and deaths of early August this year, which shows that the majority of cases and deaths in Texas were male. In addition, the majority of cases range from 20 to 59 years old, while the majority of deaths were 80 years and older, with people ranging from 70 to 79 right behind. And lastly, in relation to ethnicity, Hispanics made up of the majority of cases and deaths in Texas, with Caucasians in second place in both categories.
When comparing the information from California and Texas age data, we concluded that in Texas, the majority of infectious cases was between 30 and 39, while in California, it was between 18 and 49. But in deaths, Texas was leading, the age group was leading with 80 years or older, while in California, it was 65 years or more. Oh, can you go to the next slide, the back one? While the gender data was, the majority of cases in Texas were male, but in California, it was evenly distributed between the two genders. So there was no preference between any of them. And in deaths, the majority of cases were in both states were males.
And then for our ethnicity data, majority of the cases in Texas and California were Latinos, along with the native Hawaiian and other Pacific Islanders living in California. As for deaths, the majority of deaths in Texas were also Latinos, while there was no majority ethnic group that died in California.
Next slide, please. Our comparative findings found that with all of our categories, our genders show that more males have died compared to females, as shown with CDC's findings. And as well for ethnicity, we have that in local areas, more Latinos have died, whereas CDC found that overall, we have more African Americans dying from COVID-19. Next slide, please. Lastly, we would like to thank Mr. Ni Tran, Professor Wei Sin Yao, our peer mentors, and the following donors for their generosity and guidance.
We would like to thank Ms. Rochelle Campbell for her generous donation.
Dr. and Mrs. James Whitehead from the CNAS RISE program.
And Mr. Washi Ahmed from the class of 2001 CNAS program.
Thank you so much for joining us for our presentation today. We're now open for questions.
All right, thank you very much. Great presentation, everybody. And yes, great teamwork. I do like to comment that you guys all did a wonderful job for jumping into a statistics, physical sciences laboratory and research project, despite all being life sciences majors, except for Alexandria, sorry. You are in fact a statistics major, so this is probably, again, right within your field, and a great job, really great teamwork from everybody. If anybody has any questions for our panelists, please feel free, type in the chat. And there is a virtual hand. You guys can raise that. It is possible. And we can unmute you to ask a live question if you would like. Okay, looks like Marissa does have a question. What was it like working on a subject or this data that is so relevant for today?
It was very interesting being not from like a stats background. I struggled personally at the beginning. I was just really bummed out because I wanted to be in a fruit fly lab or like something like butterflies or lizards, and having to just do computation sounded boring. But I think the fact that COVID-19 is of greater relevance today, it constantly motivated me. And being in a science field, you want to be, you strive to make a difference. And I feel like even if these findings might change because the virus is constantly mutating, I think what we did here was the first step into a door with more solutions to this global pandemic. So it was really it was really nice to be able to be one of those people making that difference. So that's something that I enjoyed.
All right, you guys did get another question. Was there any data on children? And can you elaborate on that?
Yeah. Nicole, what was the age range, Nicole? 0 to 18?
So actually, yes, the age range was, we had categories from 0 to 17, which would include children. And as we saw in the slides earlier, for positive cases, 0 to 17, 50 to 64, 65 and older, they all had like a similar trend line of growth. But for daily deaths, children have died at a much slower rate compared to the elderly. So that was the data we found from the California database.
All right. Well, thank you very much. And again, thank you again for a great presentation and working together, all three of you. We will be moving on to our next presentation.
Thank you, Nate.
Thanks.
All right. So our next poster is, actually not poster, oral presentation is now on the board here. And then we will have Ms. Radhika Amin. Go ahead and you can start whenever you're ready.
Hi everyone, my name is Radhika Amin and I am a second-year neuroscience major at UCR. Today I'm going to be discussing the project I was working on during RISE and in conjunction with the UCR Brain Game Center. The project involved data collection from a cognitive battery that will be administered longitudinally to stroke patients. Next slide, please. So first and foremost, before delving into the specifics of my particular research topic, I would like to go over what a cognitive battery is in the first place. So it's basically a group of assessments that can test for multiple cognitive domains, such as hearing, orientation, attention, and executive functioning, so forth. And the way that we determine which cognitive domains will be included in a battery is dependent upon the target population. So for example, there can be batteries designed for people with autism spectrum disorder, ADHD, schizophrenia, and so forth. So in my particular case, since we are evaluating a target population of individuals affected by stroke, we want to choose domains that are typically affected in stroke patients, and that also goes with what type of assessments we'll be choosing. Next slide, please. So this is the tentative timeline of our project. During summer of 2020, we wanted to determine which domains and assessments we would include in our battery. During fall of 2020, we hope to develop and validate the battery and possibly pilot it to our target population. During the winter of 2020-2021, we hope to administer the battery to a wide population of not only stroke patients, but also healthy individuals. To sort of delve into what I did during RISE, we examined the literature, and in doing so, we learned which impairments commonly presented in stroke patients. Then we determined which domains we would like to include in our battery, and for the last step, determining the assessments, we're still working on that because we want to make sure that we have the right assessments before compiling them together in the battery. Next slide, please. These are the two main aims of our project. So first and foremost, we would like to develop and validate a neuropsychological battery designed for stroke patients. So first, it has to be remotely administered, sensitive, electronic-based, and have the ability for long-term administration, rather than administration at just one point in time. And also to kind of go over what we mean by electronic-based, we want to have computerized versions of these tests, as they are more specific than the paper and pencil versions that are commonly offered to stroke patients. So that's something I'll kind of go over later in the presentation. Second of all, we would like to determine the cognitive profiles of stroke patients. And cognitive profiles are basically patterns of performance on neurological assessments. So that's basically what the battery is doing. So we'd like to build the database, as I mentioned earlier, from stroke patients and also a healthy population, and then we would analyze the data to see what the patterns are. Next slide, please. So why do we need to create a neuropsychological battery in the first place, and why are we doing this research? So first of all, existing stroke batteries such as the NIH stroke scale, the MMSE, and the MOCA are not designed for to be widely administered nor available. So there are logistical difficulties in these batteries because experts have to be trained. It's costly, expensive, and also these examiners may not be able to administer the test to stroke patients as they might not be available. And on top of that, the existing batteries miss important functions, as the MMSE has been cited to miss several important executive functions, which is not good. Second of all, patients in the existing batteries, they are designed to assess patients at one point in time, such as admission into the hospital, rather than over time. And as you can imagine, the cognition of stroke patients is very, very rapidly changing and evolving. So we want to see how their cognition changes over time. And third of all, we see that when we do not screen for do not formally screen for impairments in stroke patients, this leads to the impairments being untreated, which can lead to poor quality of life and also poor long-term outcomes. And the literature has shown that when patients were not screened with a battery, 35% of them had 3 or more undiagnosed cognitive impairments, which is definitely not good for the patients. Fourth, given the current coronavirus situation, this highlights the need for remotely administered tests, as expert examiners and other sort of experts may not be able to enter the hospital and administer these tests as they were once able to. Next slide, please. These are some common domains that are tested by stroke batteries, and I'm going to be going over the ones that we plan on including in our battery. So first we have executive function, and under executive function, we would test things such as motor speed, motor function, and also other things such as inhibitory control, cognitive flexibility, decision-making, and so forth. Working memory is a process involved with storing memory in the short term to be used, and also pulling information from long-term memory so that it can be used. So some things that we would test under working memory would include recognition, recall, and delayed recall. Visuospatial function involves using cues, spatial cues in one's visual field in order to detect and see certain objects. So for example, a patient who presents with visuospatial neglect may not be able to see something in their left visual field because they have a lesion in the right side of their brain. So this is something that we would like to investigate in patients as well. Next slide, please. These are some domains that are not commonly tested in stroke batteries. So we have hearing, vision, and mood and emotion. And it is important to note that all 3 of these domains have been shown to be predictors of long-term outcomes in patients. So that is something worth looking into. But we personally decided to test hearing and vision in the battery. Next slide, please. So I'm just gonna go over 3 tests that we plan on including in the battery, and it's not finalized yet, but these are just some examples. So first we have the Trailbaking Test, and it mainly tests the 2 domains of executive function and motor function. And the participant is required to draw a line to connect the circles in a certain order in parts A and B, as you can see on the right side. The main outcome of the paper and pencil version of the test is just the time used to complete parts A and B. But with the computerized version, we can gain more types of dynamic data and main outcomes, which are things that we measure. So that can include total pauses and lifts, average pressure the participant used, and the time spent between circles. And these additional main outcomes can give us more clues into a patient's cognition and see how they are progressing over time. And I would like to add that the Braining Center is working on developing and validating a computerized version of the Trailmaking test, so we hope to use that in our battery. Next slide, please. This is another test that we intend on using. It is called Spatial Release from Masking, and it mainly tests the domain of hearing, as participants are required to use spatial cues in order to detect sounds in their field of hearing. So the participant is required to select a specific color number combination based on a call sign. And the call sign is a name and a color number. So for example, the participant may hear Charlie, go to blue 4 now. And as you can see on the right side of the screen, they would have to select blue 4. The difficult part about this task is that there are multiple voices coming from different areas in their hearing field. So they have different call signs. So for example, they might hear Mary, go to red 3, but their goal is to find Charlie to the best of their ability. The main outcome for this test is just correct responses. And I would like to add that the Braining Center has widely used this task, and it has been validated and developed on mobile platforms. Next slide, please. This is another task that has been developed and validated by the Braining Center and used widely on mobile platforms. It's called Cancellation Pictures, and I would like to add that cancellation tasks are widely used in the stroke literature, but they are mainly paper and pencil tasks. And they're used to determine how patients are seeing objects in their visual field. So the main domains that this cancellation picture task will test are visual spatial function and executive function. And this task is based on a paper and pencil cancellation task called the D2 task of attention. So the participant is required to select the targets that are in the correct orientation, as you can see on the right side, and they are required to do it as fast as possible. The main outcome for the paper and pencil version of the task is merely accuracy, but with our computerized version, we have the option of gaining additional types of data, such as reaction time, which is a type of executive function. So we hope to use these additional types of dynamic data to ascertain more about how a patient is progressing. Next slide, please. So we hope that in the future, the battery may be used to determine the efficacy of certain stroke treatments. So there are many types of stroke treatments. For example, the cognitive battery could be used in a drug trial to see how a specific drug is affecting a patient's domain, and a specific domain, such as hearing or vision or executive function. And it could also be used in rehabilitative therapy to see how the therapy is affecting the patient's cognition as well. Next slide, please. So I'd like to thank my PI, Dr. Aaron Seitz, our lab coordinator, Audrey Carrillo, staff and students at the Braining Center, and everyone who was involved with RISE, the coordinators, the peer mentors, and also the John and Elizabeth Leonard Family Foundation, who has funded my research. And without all of these individuals, my project definitely would have not been possible. So I'd like to thank all of these individuals so much. Next slide, please. I'd like to thank you for listening to my presentation. I'd be happy to hear of any questions you may have. And on the next slide, I have some references.
All right, thank you very much, Radhika. Great presentation, that was extremely interesting. Does anybody happen to have any questions? Please feel free to type in the chat here or raise your virtual hand. And since you were actually part of the program prior, I think I remember asking, did you get to experience working with this lab before the summer?
Yeah, so that was actually really interesting because I've been working in this lab since last fall. So I had a very thorough experience of research in person. And also now I have research experience online. And the two are very different, but I'm really thankful that I was able to do both and experience both, like even from a distance. And my favorite part about the research project was, I think it was really interacting with many different experts in the field, like working with the doctors at the UCR School of Medicine, working with my PI, Dr. Aaron Seitz, and all of these experts who really know so much about stroke, and also working memory tasks and computerized tasks for psychological well-being. So really working with like all these different individuals and trying to pull that together to form our holistic battery was probably my favorite part.
Awesome. Thank you very much. Okay, so we'll be moving on to the next portion of this second session of the morning. We are transitioning into poster presentations. And so the difference for these presentations is students were requested to make an actual poster, and we will have a zoom function into the specific areas that they will focus on. Timing is also different. It's a little bit less. They're giving 8 minutes total. So they will be presenting for approximately 6 minutes with a couple 2 minutes or so for questions at the end. And our first poster presentation is going to be Ms. Michaela Hendry. If you get opportunity, please turn on your camera. Thank you very much. And your poster is up. Marissa is in the background, and you can take it away.
Okay. Can you hear me?
Yep, we hear you fine. Thank you.
Okay. Hello, everyone. I'm Michaela Henry, and this summer I've been working under Dr. Subir Ghosh's lab in the statistics department, and we have been researching the COVID-19 in the United States. The coronavirus has been known to be in existence since December of 2019 and shows no signs of stopping anytime soon in the US. Our objectives are to see how this pandemic has evolved, to generate data, and to describe changes in the number of new COVID-19 cases over these past 6 months. This problem is cared about because the coronavirus affects everyone, and we think this is important because the media is constantly mentioning the phrase flattening the curve, and we want to explain what that means. Flattening the curve in the statistical sense is in relation to the probability density function, or PDF, of a uniform distribution. The PDF of a continuous distribution is defined as the derivative of the cumulative distribution function, or CDF. The CDF is the probability that a random variable takes a value less than or equal to X, and a random variable is a function from a sample space S into the real numbers. Let's zoom in on figures 1 and 2. Okay. If we compare these graphs showcasing the PDF of a normal distribution and the PDF of a uniform distribution, we can see that the graph of the normal distribution is shaped like a bell curve, and the graph of the uniform distribution is shaped like a flat line. Although the uniform distribution here is from 0 to 1, it can also go from -6 to +6. The uniform distribution stays at a constant level over time, and if we follow the line, we can see that the density will be flat. Relating this to COVID-19, if the number of cases increases or goes up, the curve will also go up. If we make this level a lower value, this also affects the height of the density, and the distribution will become uniform because the number of cases is not increasing. And of course, a lower height is better. Let's zoom back out. So to get our results, the methods used were graphing and data analysis. First, I went to the Our World in Data, or OWID website from the Global Change Data Lab, or GCDL, in the Oxford Martin School at the University of Oxford to find data for new cases of COVID-19 from the months of March to August. Then, in Microsoft's Excel, I organized the data with every 2 columns representing a month of new cases. In a separate Excel sheet, I made each pair of columns into a table where columns represented the days of the week and rows represented May and June. I calculated the average numbers of new cases per day of every month and the average number per week for May and June. Finally, I took the average of each month. Several line graphs were plotted and a histogram was made with this information. Let's zoom in on figures 3 through 5. From these graphs, it can be concluded that there is a big increase in cases from March to April, no big jumps from April to May to June, another big increase from June to July, and a decrease from July to August. Although there was a decrease, the US isn't from COVID-19 yet and still must be careful. No complete analysis can be done on the month of September because obviously the month isn't over yet. March has shown the lowest number of average new cases per day, and July has shown the highest. The implications and the importance of these findings are that if the numbers for September and October decrease, the worst of the coronavirus may be over, that state restrictions are working, and that the US may be starting to flatten the curve, although it still has one of the highest rates of infections in the world. It appears that the US may have already reached its peak with the coronavirus according to these graphs. Let's zoom back out again. The next steps in our research are to get these kinds of graphs with average new numbers of COVID-19 deaths and hospitalizations, and to figure out the type of statistical distribution from these plots. The plan is to estimate the CDF and PDF of the graphs, and then try to predict what will happen next with the coronavirus. Now I'd like to take some time to properly acknowledge all the people who made this presentation possible. My PI, Dr. Subir Ghosh, my lovely peer mentor, David Kisse, and co-peer mentor, Gina Duo, my donors, Mr. Kenneth and Nancy Gray, my mother, the CNAS Connections Program, and the University of California, Riverside. Thank you all for supporting me on this project.
All right, thank you very much, Michaela. I think despite the slowdown in transmission for Zoom, we were able to make out Michaela's final acknowledgements to the CNAS STEM Summer Bridge Research Program, as well as UC Riverside. And it looks like she may be frozen. And that's, to be honest, that's a pretty good picture, smiling picture to be frozen on. All right. It's like we may have lost her, which is okay. Our time is actually up for her particular presentations. We will be moving on to our next presenter, Mr. Devante Kane. Thank you very much for turning on your camera. Good seeing you, Devante.
Hi, Tony.
All right, we're doing great. And then, yeah, your poster is up and running. You can take it away.
Okay, so hello everyone. My name is Devante Kane, and my research this summer was on the convergence of discrete time random walks to Brownian motion. And so a little bit about Brownian motion is it was discovered by a botanist, Robert Brown, by taking pollen from flower and suspending it in water. And what he observed underneath the microscope was that the pollen was actually in continuous random motion. And so one of the first people to really conquer this and model this system correctly was Albert Einstein. And how Albert Einstein viewed this was that the reason why the pollen was in continuous motion was because it was in collision with the molecules from the water. And so he realized that this was actually a problem of thermodynamics, which is the theory of kinetic and molecular theory of heat. And so he was able to take that viewpoint and also model the system probabilistically, that the pollen should be in a certain interval at a certain time, and that that value should be a probability. And so by combining these two ideas, he's able to accurately depict Avogadro's constant, and he's able to derive the diffusion equation probabilistically. And so this was kind of the very first thing that I did this summer, was to go through his work and how he did this. The next thing that I looked at was random walks. And so what a random walk is we're modeling the same system one-dimensionally, and how we view it is that at a specific time, so meaning if my time step is 1 second, that means at 0.5 seconds, I haven't jumped. At 0.7 seconds, I haven't jumped. But once I get to 1 second, I jump a distance of delta. And so a random variable that models this equation will be mean free, and it will have a variance that should be a constant. And we know that the variance should be a constant because as we observe the pollen randomly moving, it randomly moves, but it randomly moves in a pretty decent sized interval, meaning that it doesn't just shoot off through the liquid. It randomly moves, but maybe in like a discrete small volume. It's contained. And so we know that the variance of the system should be finite. And if we look at the variance, it goes as del squared over tau m. And the limit of this is the diffusion coefficient. And so any system that can be modeled and has a variance whose limit goes like del squared over tau will be Brownian motion. And so this is kind of interesting. What I used to think about this was that it kind of tells us something about the relationship between space and time. But in actuality, this is true over other mathematical systems. And for example, if you use it in the p-adic setting, it is also true. And it tells you more about the relationship of how we've modeled the random variable than it does about space and time. Another really cool thing that I've looked at this summer was that if you look at the pollen molecule and you think about it from going from point A to point B, there's actually an infinite amount of ways that it can go from point A to point B. And each one of those paths has a certain probability associated with it. And with that probability comes a measure. And the measure is actually what tells you, it's a mathematical concept, but it tells you something physical about the system. And so that's what I thought was really cool this summer. In the future, what we're trying to do with this project is we're trying to model or be able to solve the Feynman-Katz integral. And we think that one of the results from our research will give us a good starting point at a difference equation that will be able to solve the Feynman-Katz integral. And once we're able to do this, we want to look at the Feynman-Katz integral over the p-adic setting and see if it leads to something that's reasonable. And so this is kind of where my research is heading, and hopefully by the end of next year, I will have published a paper with Dr. Weisbart, and I'll have my name on a paper. And so I would like to thank UCR, specifically the College of Natural and Agricultural Science and the Physics Department for accepting me as an undergraduate physics major, and as a because I transferred in 2 years ago. And I would like to thank CAMP. I would like to thank Nee Tran, Dr. Brandon Brown, Dr. Richard Cardillo, and I would like to thank Marissa as well. I would like to thank RISE, the RISE program, Dr. Connie Newton. I would like to thank Dr. David Weisbart for being an awesome mentor, and I would like to thank my family for all of their support and love. And I open up the floor to questions.
Okay, thank you very much, Devante. Very great presentation and a good way for semi-close out our morning session here. Does anybody happen to have any questions for Devante? Please chat or type it up in the webinar, or you can ask him directly. And I know, Devante, you were actually involved in Dr. Weisbart's lab earlier during the academic year. How is that transition like from actually working in the lab and doing this research to now working solely from home?
Well, because it's math, it's pretty much the same. Everything we do is, doesn't have to involve a lab. Dr. Weisbart recently published 7 papers and finished his tenure. So he's a lot more free now to meet with me, and we've made a lot more progress. And I understand the problem a lot more than when I first started a couple quarters ago.
All right.
I have a question here from Josh saying, what is the p-adic setting? I don't want to give you a wrong answer, but that is a graduate level concept. I just know that it's not the real numbers. It's a different type of number system. And that's all I can say on that. I don't want to say anything that's false.
Okay, any other questions for Devante? Okay, well, thank you very much, sir. We will be moving on. We've actually had a slight change in our schedule, and therefore we will have an extended break and start right back up at 11:30 instead. So you guys will have a little more time to go take a break, run to that restroom, grab a snack, a drink, and definitely at the minimum, please stand up and stretch. We're gonna start our timer for 13 minutes, and we will see everybody back at 11:30 sharp and start with our next and final rounds of poster presentations. Thank you very much. Okay, welcome back, everybody. I hope you had a restful and productive break. We will continue with the last leg of our second day for the RISE Symposium, and we'll continue with our poster presentations. So our first speaker for the session is going to be Ms. Natalie Tajeda. Thank you very much for turning on your camera. Your poster is up and running. Marissa is hiding in the background, and the floor is yours.
Okay. Hi everyone, I'm Natalie Tajeda, and I am a third-year neuroscience major. And for my RISE project, I have the privilege to work with Dr. Annie Dida and Dr. Rachel Wu. Specifically, these past 8 weeks, we have been focusing on learning and motivation in younger and older adults. Now, there's an ongoing discussion of general education classes. Many undergraduate students often lack the motivation to learn in these classes. This has happened to me as well. However, having a low motivation in these classes can lead to a low learning outcome, and this is not well. So we all know that having a well-rounded education can help us in our careers. Therefore, having more motivation to learn in these classes is very important. Previous work has shown that motivation is affected by several different factors. This is due to student beliefs, self-efficacy, autonomy, and other cognitive theories. But these theories do not go into the process of learning itself. Prior work has shown that knowledge and gaining knowledge through the process of learning can lead to curiosity and interest. Therefore, it creates a reward learning framework where people are more desired to fill in the gaps in their knowledge. Therefore, learning can encourage new learning. My mentors and other staff members in my lab have worked on this study this past year that demonstrating that the exposure to new information through TED Talk videos can increase motivation to learn. If we look on the graphs on the lower left corner, we can see that before the video, motivation did increase. However, general motivation remained constant or did not increase. Now, if we go back up to the research question, past research has shown that older adults tend to remember information. They're motivated to learn. However, younger adults tend to remember information regardless if they're interested in that material. Therefore, my research question is, will motivation increase after exposure to new information? Older adults may lack the motivation to learn something they are uninterested in. However, we predict that the lowest motivated topic will increase after material exposure, but the highest motivated topic will remain constant or it will not decrease. Now, if we go to our methods, after we had gotten IRB approval, I had created a Qualtrics survey. Qualtrics is an online programming survey, and I created this. Can you go a little bit up, please? Thank you. After recruiting participants through email and through phone, for instance, I called many senior centers. I started to schedule participants. I had to ensure they were 60 years and older and that they spoke English because I needed to understand and fully engage in the video in the TED Talk topics. Then I began to run participants through Zoom. Participants needed to share their screen, and this took approximately 30 minutes, and participants were compensated for their time. Now, while I was running participants, after their consent and the debriefing form, I began to assess their demographics. I asked for their gender, their age, their location, because we opened the study internationally. I asked for their ethnic and racial background, their occupation, their highest level of education, and if they had any majors or multiple majors. Then I moved on to a personality test. This consisted of 10 personality questions that went as, I see myself as an extroverted person, or I see myself as an anxious person, because we can see that motivation is affected by personality. Then participants moved on to rate their motivation to learn in these TED Talk topics. The scale depended from 1 to 9, and then they moved on to rank their motivation to learn from a scale from 1 to 6. Then they rated their knowledge on each of the following topics from 1 to 9. Based on these results, Qualtrics was programmed to either give them the lowest or the highest motivated topic. The videos ranged from 14 to 22 minutes. Then we moved on to rating their motivation to learn after the video. Now, if we look at the bottom, we can see the layout of the surveys. Thank you. In the figure 2, we can see participants rated their motivation from a scale from 1 to 9. 1 being not motivated at all, to 9 being extremely motivated. The topics consisted of cell biology, civil engineering, computer science, entomology, mathematics, and physics. We then moved on to ranking, from 1 being their top choice to 6 being their lowest choice. Then they rated their knowledge on the following topics, from 1 being having no knowledge at all to 9 being an expert in their field. Now I'm currently collecting data. Can you zoom out, please? I am currently collecting data through Qualtrics and Excel. However, my next steps is to continue to run participants. We want to run at least 40 paid participants. And after collecting all my data, I'm going to assess statistical significance. And we hope to repeat the study with a different population. We have done it with undergraduates, now older adults. The next step is to do it through middle-aged adults of the age 30 to 50 years old, because we want to see how learning and motivation is affected across the human lifespan. Thank you everyone for listening to this presentation. I would like to thank my mentors, Dr. Annie Dida and Dr. Rachel Wu, for helping me and mentoring me and the guidance. I would also like to thank all the CNAS Persist staff, Gina, Jen, and Darren, Nee, and Marissa for their constant support and for making RISE possible. I would also like to thank Elizabeth Fletes for being my mentor throughout this entire process, guiding me every step of the way. And I would also like to thank the National Science Foundation for funding my project. Thank you, everyone. I now open the floor for any questions.
All right, awesome. Thank you very much, Natalie. That was a great presentation. Dr. Brandon Brown has a really brief question for you as well. He'd like to know, how do you feel about learning and motivation? How do you feel learning and motivation has changed during the COVID-19 pandemic?
It has definitely changed. Many people are discovering the new learning methods, myself included. I've learned that different things don't always work out. So it's very interesting to see how learning is affected during this time period. Oh, thank you everyone for the nice comments.
All right, thank you very much. Well, we will go ahead and move on to our next presentation. Thank you, Natalie, great job. And Ms. Sarah Cabarra, thank you for turning on your camera. Your poster is up and running, and the floor is now yours.
Hello, can you guys hear me? Are we good? Okay.
Yeah.
Hi, everyone. My name is Sarah Cabarra. I am currently a neuroscience major working under Dr. Aaron Seitz in the Department of Psychology in his Computational Neuroscience lab. And our current focus is on working memory. So first off, what is working memory? If you can go to the introduction section of my poster, please. Okay, so working memory is a cognitive ability that is related to certain things such as reasoning, decision-making, and generally goal-oriented behaviors. It is it involves the retaining and manipulating of information to perform tasks successfully. And so our goal in my lab is to boost the effectiveness of working memory. Now, why do we care, right? So working memory is very demanding. It can also be very impaired in certain people. So for example, somebody with a learning difference, such as attention deficit disorder or ADD, often suffer from impaired working memory. And current approaches to engaging working memory and just current approaches to learning in general are often deemed very challenging because they typically overload executive function. And the reason why this makes it challenging is because working memory has a very limited capacity. It can only hold information temporarily. So when you overload executive function, it makes it very difficult for certain individuals to focus and to effectively retain and manipulate information. So my lab's approach to engaging working memory is essentially tricking the brain into learning. And I'll get into the hows in a minute, but I need to define a few other things first. So our paradigm has a basis in what's called task irrelevant learning, which is a phenomenon where a stimulus that bears no relevance to a task can still be learned due to its consistent presentation during the task. And so essentially a stimulus that can give no useful information to the completion of a task, but it can still be learned just due to how many times it shows up during the task. And additionally, we will also be using the release of norepinephrine to help facilitate learning. And for those of you who don't know, norepinephrine is a neurotransmitter associated with many cognitive functions such as attention, learning, and memory storage. And the main reason why we're looking at norepinephrine is because recent studies have shown that when a stimulus is presented during the release of norepinephrine, the stimulus is more likely to be learned and retained. And this is regardless of the inducing stimulus. And so when I say this, okay, let me give an example. So stimulus A it triggers the release of norepinephrine, but stimulus B is present while norepinephrine is released. Stimulus B will be learned over stimulus A due to the fact that it was present while norepinephrine released, despite stimulus A being the trigger. And that's very important for our experimental design. And so we plan on modulating the release of norepinephrine with what's called novel or oddball sounds, which have been shown to trigger the release of norepinephrine. I will be using the words novel and oddball interchangeably because they essentially mean the same thing. So when I say tricking the brain into learning, I essentially mean using the release of norepinephrine to induce learning. So if you can go to the methods, please. So like I said before, our paradigm has a basis in task-relevant learning. We will be using a visual end-back task with paired auditory sounds. For those of you who don't know, the end-back task is a well-established method in assessing and measuring working memory. It typically consists of a sequence of stimuli being shown, and the subject will be instructed to indicate when the current stimulus in the sequence matches a stimulus shown previously a specific amount of steps back. And so N is like the manipulating factor. You can manipulate the difficulty of the task. So you can do 1 back, which is easier than say 3 back. And so while this design has yet to be finalized, I can give like a general overview of what we might expect for the SIGU GOAS taken from previous studies. So if we direct our attention to the figure to the right, the leftmost green circle is our target visual. This is what we want to be held in working memory. And we will be pairing it with an oddball sound, which is indicated by the filled-in speaker. So this is actually an example of a 2-back condition. So 2 steps later, we see the green circle appear again. In this instance, the subject would indicate that the current stimulus matches the target visual 2 steps previously. And so this paradigm will be coded and executed by the Inquisit software. And while this is still in the finalization phases, we are still anticipating certain results, and we are also asking how we could possibly analyze data. So if we can go to the graph below in the resulting data analysis. Yeah, okay. So in terms of what we expect to see, we are just essentially expecting to see that the participants successfully complete the end-back task. This would mean that the target visual that is paired with a novel sound has been successfully retained in working memory. But we are also looking for efficiency and alertness being maintained throughout the task because we are designing this paradigm to not overload executive function. So ideally, all attention should be focused on the visual end-back, which is the primary task. And in terms of how data will possibly be analyzed, we can look at target accuracy and reaction time. So, and what I mean by target accuracy is that target visuals that are paired with novel sounds should have higher accuracy than non-targets. And this is because, of course, we are pairing it with a novel sound. And so for reaction time, it's the same kind of concept. Target visuals should have lower reaction times because they are paired with novel sounds as well. So the graphs below are actually taken from a previous experiment that had a similar kind of experiment design. Instead of using an end-back, they use a scene recognition task paired with novel auditory sound. And so it basically went with a target scene being paired with a novel sound, then shown in sequence with distractor scenes and blank screens. And then after the sequence is shown, the subject would be instructed to indicate if the target scene showed up in like this little miniature quiz after the sequence. And so as we can see, the left graph is accuracy. And so we see that novel accuracy had an increasing trend versus the repeat accuracy. And on the right, which is reaction time, novel screens had lower reaction time than the repeat screens. So with all this said, this project has yet to be finalized, but this of course just means that the future steps is to finalize the project, create the standard operating procedure, and then finally recruit and screen participants. And with all that being said, I really am really excited to be a part of this project. I have never worked in working memory, in general, and I would really like to thank my graduate student, Lhasa and my PI, Dr. Seitz, for giving me all of the assistance and advice during this period. Additionally, I really like to thank Dr. Brown, Tran, and the CAMP program for giving me this opportunity to conduct research over the summer. And of course, Isaiah Grant and Karina Morales for being amazing peer mentors. And then of course, the NSF for this funding opportunity. Thank you so much for listening. Does anybody have any questions?
All right, thank you very much, Sarah. Great presentation. Very smooth, and looks like you do have a question from Ms. Sally Kabara.
Okay, if you're talking, I can't hear you. For some reason, what's it called, just stopped working.
No.
I can't hear anybody right now. I'm sorry.
No worries. Can you hear me now?
It's not working right now. I don't know what's wrong. Okay, well, this is kind of embarrassing.
it is okay, then we can type in. If you want to, if you can see, or if you can
Yeah, I
happen
can't hear
to hear.
anybody. How many
No.
people have you asked to trick their brain into learning? Please trick mine. We haven't asked anybody yet. I will probably be one of the people who will have their mind tricked into learning, because undergraduates are kind of forced to be in the projects. But I don't mind. I will, if you want to be part of it, I will definitely include you as well. Okay, so there's another question. When you refer to learning differences, do you mean learning differences like dyslexia? Yes, that is included, because they often suffer from impaired working memory as well. And that also leads to their impaired focus, makes it difficult for them to focus on other things as well. I still can't hear anybody, but yes.
It's okay. We're going to move on to our next presenter. Thank you very much, Sarah. And yeah, great presentations. You did great. You still were able to answer everybody's questions, and as they're typed in. So our next presenter will be Mr. Daniel Avalos. Thank you very much.
Can you hear me?
We hear you perfectly. Your poster is up and running, and you can take it away.
Okay. All right. Hi, my name is Daniel Avalos. I worked in Dr. T.Y. Woodruff's lab, where I counted the number of demyelinating myelin-making cells in a demyelinating cerebellum after cuprizone dieting in mouse models of multiple sclerosis. So let's begin with the introduction to multiple sclerosis. Multiple sclerosis, MS for short, is demyelinating neuroinflammatory autoimmune disease of the central nervous system that affects neurotransmissions across the central nervous system. Marissa, can you please zoom in on figure 1 of the introduction, please? Produced by oligodendrocytes, myelin helps axons transmit neural signaling. However, MS activates an autoimmune response by sending lymphocytes to destroy myelin sheaths around axons or directly attack oligodendrocytes. This decrease of myelin, demyelination, results in distorted neural signaling and consequently end in neurodegeneration. Marissa, you can zoom out. MS is a multifarious disease of the central nervous system. However, it commonly impairs cerebellar activities. The cerebellum is important for coordination, regulating body movements, and motor learning behavior. Cerebellar dysfunction as a result of MS include walking abnormalities and tremors. To study the stages of demyelination with MS with demyelination in the cerebellum, cuprizone diet mouse models were utilized. For this lab, we used PLP EGFP mice to target CC1 and OLE2 in white and gray matter regions of the cerebellum. CC1 is a protein marker for mature oligodendrocytes, whereas OLE2, it's a transcription factor gene expressed in all oligodendrocytes. Cuprizone is a copper chelator that's toxic to mature oligodendrocytes. Therefore, I hypothesized that over time with the cuprizone diet, we will see fewer mature oligodendrocytes in the cerebellum. Moving along to the methods, PLP EGFP mice were received from the Jackson laboratory. Marissa, can you please zoom in on figure 2? Yes, okay. The mice were set in 2 groups, a control group that was fed with a normal diet and an experimental group that was fed with 0.2% cuprizone diet for 3, 6, 9, or 12 weeks. At those weeks, the mice were perfused and their cerebellums were processed for immunohistochemistry, IHC for short. Marissa, you can zoom out. IHC was performed to stain oligodendrocyte markers OLE2 and CC1. Marissa, can you please zoom in on figure 4, the one with the red oval? IHC staining utilizes antibodies labeled with fluorescent dyes to visualize markers in tissue samples. The target molecules that we're after are CC1 and OLE2. Marissa, you can zoom out. Image J was used to count fluorescent markers that represent myelin-making cells in white matter regions of the cerebellum. In figure 5, I processed these black and gray images shown below using image J. I outlined where the white and gray matter regions are in the cerebellum. I also counted the number of myelin-making cells in each region. Bright white shapes I identified as a cell. What we found in our research, Marissa, can you please zoom in on figure 6 in the results, the glowing picture, please? Yes. The image shown is the cerebellum. I counted the cells on image J in grayscale, but when the images in figure 5 are combined, it becomes colorful, as shown in figure 6. As you can see, DAPI cells, which are blue, DAPI stains the nucleus of neurons. CC1 is colored red, and OLIG2 is colored green, which are found in white matter of the cerebellum. In comparison to the normal control group, we found a significant decrease of CC1 cells in the white matter region of the 12-week experimental group. The reason why is because cuprizone disrupts mitochondrial activities needed for lipid and ATP synthesis. OLIG2 are found in precursor oligodendrocytes that can differentiate, making it less prone to degenerate. Whereas CC1 found in mature oligodendrocytes, they lack this ability, thus resulting in degeneration. Marissa, you could zoom out. Thank you. The advantages of using cuprizone modeling is that it's reproducible and amicable for studying the pathology of demyelination. The disadvantage of cuprizone modeling is that MS is an autoimmune disease that directly damages myelin sheaths, whereas cuprizone induces degeneration of oligodendrocytes and not attack myelin sheaths. Considering future studies, we look to other comparisons in experimental groups of week 9. We also look forward to study gait and other motor learning regulators to test the possible restorations of these cells. In closing, I would like to say to join the research. Kelly Atkinson, one of Dr. Tiwari Woodruff's research graduates, for taking me under her wing and helping me during the research. I would also like to thank Dr. Ford, Dr. Herrick, and Dr. Wolde Ab for the introduction to undergraduate research. And a big thank you to the NIH, National Institute of Medical Sciences, and of course, Bridges to Baccalaureates for funding this experience. It's an honor and privilege to be here. And now I'm opening the floor to any questions.
All right, thank you very much, Daniel. Great presentation. You do happen to have one question here from Marissa. How quickly does the cuprizone demyelinate the axons once introduced into the diet?
Okay, yes, so usually demyelination, from what I understand, begins around weeks 1 through 4, but it becomes more prominent during weeks 5 and 6. And then the point where remyelination of these axons become a failure, I guess, are in weeks 9 through 12, or however longer you induce the cuprizone diet.
All right, and you happen to have one more question from William. What other applications do you think this research could have in studying chronic neurological conditions and diseases in humans?
Okay, so like I mentioned in future studies, we would like to analyze gait and other motor learning functions. So gait is just basically the strides of the subject's walking abilities to see whether their coordinations are impaired or not, and other motor learning behavior. So this could be like limb coordinations and so on and so forth.
Awesome. Thank you.
I hope that
much.
answers your question. Okay.
Well, they will have to wait. We're going to move on, but thank you very much. Great presentation.
Of course. All right. Thank you.
All right, Mr. Michael Asaf, thank you for logging in. Your poster is up and running, and Marissa will guide you on the back end. So please take it away.
All right. Hey, everyone. My name is Michael Asaf, and throughout the course of the past month, I was fortunate enough to work with Laura Dovik and Dr. Vijay Santhakumar remotely from home. Our research question was, do changes to learning and memory in disease result from altered neuronal activity patterns? So just to start it off, the hippocampal dentate gyrus is believed to regulate the formation of memories. However, traumatic brain injuries as well as epilepsy have been known to have the comorbidity of memory deficits. The main cell type in the dentate are the granule cells, which can be further subcategorized into the granule cells and the semilunar granule cells. Now, this is what we hypothesize will also play a role in memory formation. We studied learning and memory behaviors in mice performing spatial memory tasks to examine how they are affected in brain injury and epilepsy, and to see whether there are changes in neuronal activity patterns may cause memory deficits. Now, throughout our investigation, we concentrated on behavior tasks that assess spatial memory, which was the Barnes maze. Now, the Barnes maze is basically a circular arena with 19 decoy holes, a starting chamber, an escape hole leading to a tunnel, a metal support frame, a camera, and several visual cues on black curtains surrounding the maze. Now, the visual cues play an important role for the mice in terms of learning where the escape hole is. Furthermore, both the open arena and the bright artificial light pointing at the table serve as a negative stimulus, motivating the animals to perform the task. Now, each animal is given 3 minutes to successfully discover and enter the escape tunnel. If they don't enter by the end of the trial, it is guided by the experimenter to the escape tunnel. The animal's behavior was quantified by recording the time it took for the mouse to discover the escape hole, as well as the errors it made. Each animal was tested a total of 3 trials per day for 6 consecutive days, followed by a probe trial, which is basically where the escape box is removed and replaced with a decoy box on the 7th day. The probe trial provides up to 90 seconds for the mice to inspect what was previously known as the escape tunnel. The recording followed strict visual measures to analyze 3 different data points, which is the number of incorrect decoy holes checked prior to the actual escape tunnel, the time to first check the escape tunnel, and then the time for full entry into the escape tunnel. The errors were recorded by visually assessing if the animal checks the decoy hole and are scored through manually tallying the encounters. Now, our study focuses on the dentate gyrus and the cell types involved in memory formation, as well as how learning and memory are affected by traumatic brain injury and epilepsy. Now, using a deducible fluorescent mouse line that allows us to capture the neurons that are activated during the spatial learning task, this further allows us to confirm the involvement of specific neurons in learning and memory. Additionally, using the fluid percussion injury model of rodent concussive brain injury and the pilocarpine-induced epilepsy model, we tested the effects of brain insults on animal abilities to perform the spatial memory tasks. The objective of our study was to establish a baseline for future studies looking into how granule cells' involvement in learning and memory are altered in traumatic brain injury and epilepsy. Now, if we can zoom in on figure 1, Figure 1 shows the mice data, and it portrays an improvement over time to inspect the actual exit. Now, this demonstrates the potential for improvement in memory and learning ability over the days. Now, if we focus on set A provides 2 heat maps, which describes the movement and position of an animal over time during 2 different trials. In this example, these are probe trials. The one on the left shows an animal that was never exposed to Barnes maze training compared to the probe trial with a trained mouse that successfully used its spatial cues to guide itself into where the escape box used to be. These images demonstrate that by doing the Barnes maze training, animals are able to use the spatial cues around them to identify where an escape used to be. So overall, this is a validation task to show that we are able to teach animals a spatial learning task. Now, if you focus on set B, this shows how we measure the progress throughout the trials. We analyze both the primary latency, which is the first time the animal inspects the escape hole, as well as the amount of errors it does. Now, both graphs of these trials shows a decrease in time to find the primary exit. However, the number of errors did not decrease as significantly for the second figure. This is important in terms of validating the data and proving that the Barnes maze as an apparatus was an effective method of testing the mice. Now, if we look at set C, these are samples of images of neurons that were tagged as active during the learning and memory task. What is exciting about this is that we can see both the granule cells and the semilunar granule cells tagged as a result of the Barnes maze training. What makes this really unique is that it's the first time both of these cells have been seen active together during a learning task. Now, if we could zoom out of figure 1 and zoom into figure 2, now, these 2 figures compare ability of control and brain-injured mice to perform the task. 2 months after injury, basically it did show a decline, but it wasn't significant enough. And if we look at figure 3, figure 3 shows mice undergoing pilocarpine-induced seizures that were tested in the Barnes maze 7 days after the induced seizure. Pilocarpine seizures impaired acquisition of spatial memory, as shown by the increase in time to find the exit. And in conclusion, the results from this initial data demonstrate the potential of these mice to learn and perform the spatial memory tasks in the Barnes maze. As for the traumatically brain injured, the data lacks significant difference because of the control in the traumatically brain injured mice. The results may have been influenced by the extended recovery time for the injured animals. As far as the pilocarpine versus control, there was a significant difference between both the pilocarpine control and the pilocarpine control. The activated granule cells and semilunar granule cells and neurons in relation with learning and memory are still being investigated. I'd quickly like to thank Laura Dovic and Dr. Santhakumar for their support with every step of the process. Also, I'd like to thank Dr. Ford and the Bridges to Baccalaureate program and everyone in the RISE program for making this opportunity possible. Thank you.
All right, awesome. Thank you very much, Michael. I'm gonna take a breath just for you. But that was a great presentation, a lot of information. Sounds like you did a lot of research, a lot of work over the summer. All right, the floor is open for questions. Does anybody happen to have a question for Michael? You can type in the chat, raise a virtual hand. I think still quite a bit to process. Yeah, quite a poster, putting in a lot of information there. It does show that all the hard work that you've put in.
Yeah, me, even though it was just in the time span of 2 months, we did focus on a lot of aspects during the research.
All right. Well, if there are no questions, we will be moving on to our next presenter. Thank you very much, Michael. So Fatima, we have your poster up and running, and I will give you the floor, so take it away.
Yes, thank you very much, Nee. Hello, everyone. My name is Fatima Macedo, and today I will be presenting on the effects of rhizobial inoculant for legume crops. So this past summer, I worked under the guidance of Dr. Sachs' lab, and we focused on studying the symbiotic relationship between leguminous crops and biological nitrogen-fixing bacteria. So to explain this, I put a pretty picture of a plant, as we see in Figure 2, and its roots are growing underneath the soil. There are circular growths, and what we see there are nodules. Within nodules live bacteria communities that get nitrogen from the atmosphere and create it into ammonia, a useful way for the plant to grow. So over time, there has been a lot of studies published that show there is success in using this bacteria as fertilizers and helping the plants grow. Now, our research question is, what factors influence the efficiency of rhizobia bioinoculants on legume crops? And when answering this question, our objectives were analyze and compare studies using bioinoculants and measuring for symbiotic efficiency in legumes of interest. And we had 6 different legumes that we were focusing on. And our second objective was to generate a database of studies with differences in experimental factors that evaluate the efficiency of rhizobial inoculants on legume crops. So if we can zoom into Figure 1 in the methods, this explains how we went about this meta-analysis. We began by searching studies on websites such as Web of Knowledge and Google Scholar, and then we focused on papers on studies that gave us details on the rhizobia species inoculated. This is because we only wanted 2 different species, that was Bradyrhizobium and Rhizobium. We also needed data for the total shoot dry weight and the experimental factors. If this data was given, we then further looked at the details, more details of the study, such as the soil used, the type of strain, and the plant host, as well as the geographic location. Once we have those studies on file, we kept them just in case we needed them for future reference, and then we calculated the host relative growth percentage. This compares how well an inoculated plant with the bacteria, how well it grew compared to the plants that didn't have this bacteria. If the percentage was positive, we have a beneficial bioinoculant, and if it was negative, then it wasn't beneficial. We took this data and we used RStudio to create statistical analysis and tests, and to compare and determine what were the variables that were drivers for efficiency of bioinoculants. So we can zoom out of this method, and I'd like to point out that we're focusing on 6 different legumes, but over the past 8 weeks, we didn't amount enough data for all crops. So for the data and analysis, we only focused on cowpea, which is one legume. And could we please zoom into table 1? Table 1 is the means and standard errors of relative growth for each variable measured. So this lets us know that the data we found wasn't just due to chance. We can actually take this data and further analyze it. And figure 3 is an example of how we're able to compare all that data. We have the geographical location of 3 different areas, Africa, Brazil, and the US, and we're comparing that with the relative growth percentage. So the blue line is a median line, and this shows how well the different strains did, bacteria strains. We see that in Brazil, a lot of strains did really well, and there were others that did under 0%, which was not a beneficial bioinoculant. Now we can zoom out of the data and results, but from these results, what we can conclude is that there are significant differences among host relative growth for most of the variables tested. Now, 77% of those inoculations were actually beneficial and provided benefit to the different cowpea cultivars. Now, the other were ineffective inoculations, and the pattern that we found was that they were driven by other microbes in the soil. So what we can conclude is that lower host regulative growth percentage values appear where there is more microbes in the soil, more competition, and this happens in natural soil when the soil was not sterilized prior to planting and in a field setup. So our future and ongoing work is to find enough data for the rest of the legumes in order to create these data analysis. And once we have that, we really want to find and explain what are the main drivers of success for using these bioinoculants in agricultural settings. Now, this knowledge would provide agricultural workers a lot of help in designing sustainable and natural tools. So essentially, bacteria can replace chemical nitrogen fertilizers, which pollute a lot in our environment. And to conclude my presentation, I'd like to acknowledge everyone that has helped me along the way, from those who first introduced me to what research was in the PERSIST program, Dr. Nugent and Dr. Wessler, along with Jennifer and Jaren. And over the summer in the RISE program, thank you, Nhi Tran and Marissa Carranza, as well as my mentor, Gina, and Mr. Cortez. And a very special thank you to my lab, Dr. Sachs, and my PhD student, Gabriel Barboza, who allowed me to join them and work alongside them on this project. So now I'll answer any questions if there's any, and thank you for listening.
All right, thank you very much, Fatima. That was a great presentation. And looks like you have some questions already. Maya would like to ask, what is your favorite part about doing this research project?
Yeah, so definitely it was having to look up a lot of different research papers. Prior to this, I had no experience having to look through that, even using databases such as Web of Knowledge. I hadn't done that prior to this summer. So it was having to use all those other helps. I'm also in the process of learning how to use RStudio and do all this data analysis. So I guess it was just being put in a lot of different areas, having to experience other things I hadn't done before. That was my favorite part of this project. Thank you, Maya.
All right. Are there any other questions for Fatima? Well, it looks like our time is moving along to our next presentation. Well, it doesn't show that you had too much of a disadvantage. You had a great presentation, a lot of research, and despite learning some new skills, that's kind of something we got forced into. So thank you very much.
Thank you.
All right. So we're going to have our next presenter, Mr. Stephen Johnson. Thank you for turning on your camera there. Your poster is now up and available, so the floor is yours.
Okay, perfect. Thank you. Good afternoon, everyone. My name is Stephen Johnston. I'm currently a third year studying cell molecular developmental biology. For my RISE summer research project, I was fortunate enough to work with Dr. Linda Walling from the Department of Botany and Plant Sciences. To start off, let's dive into our abstract. Bemisia tabaci is better known as the whitefly, which is an insect about half the size of a sesame seed, so super small. These insects cause worldwide crop destruction by transmitting viruses. The optimal tool to editing the whitefly's genome would be using CRISPR-Cas9. Small guide RNA lead the Cas9 complex to the targeted location in the genome. Once it is at this specified location, the double-stranded break can occur where knock-ins or knockouts are now possible. Small nuclear RNA promoters drive the expression of these small guide RNAs. Currently, there are no annotated U6 small nuclear RNA promoters in the whitefly genome. Background. The whitefly is not a model organism, so there are not many studies published on them. Out of the 32 different species, our lab chose to work with the Middle East Asia Minor 1, which is found worldwide in the host for 150 different crop viruses, as shown in Figure 1. U6 small nuclear RNA genes are highly conserved among other insects. Drosophila, which is better known as the fruit fly and closely related to the whitefly, share 97% of their U6 small nuclear RNA genes, meaning that these genes are important to sustaining life. Our lab is focused on the small nuclear RNA coded for by these genes. And our research question was, can we improve CRISPR-Cas9 mediated genome editing in Bemisia tabaci by using native U6 small nuclear RNA promoters to express small guide RNAs? In our methods, we started by comparing the U6 small nuclear RNA sequence from Drosophila to whiteflies using the National Center for Biotechnology Information BLAST database. And we did this again because Drosophila and Bemisia tabaci are closely related. Next, we aligned the two genomes and found the essential promoter elements, which are the proximal sequence element and the TATA box. Then we will isolate these regions upstream from the U6 small nuclear RNA genes. And finally, we will design a small guide RNA plasmid to test the function of these promoters. In our preliminary results, we found 5 U6 small nuclear RNA genes and their promoter. We also identified the TATA box and the proximal sequence element, PSE, and these are both needed for U6 promoter function in closely related insects. The ultimate goal of our project is to be able to insert this small guide RNA into a plasmid and be able to inject this plasmid in vivo into the organism. To be able to see if this plasmid has been successfully inserted, we will need a reporter gene. The 3 reporter genes that we tested were green, blue, and red biofluorescent protein. In whitefly embryos, they naturally biofluoresce green and blue, as seen in Figure 3. In the 3rd instar nymph stages, there was no biofluorescence. The adult whiteflies naturally biofluoresced green and blue, so this meant that DS or M cherry red would be the best biofluorescence reporter gene to use to make sure that this plasmid has been successfully inserted. Figure 2 shows the similarities between the proximal sequence element and the TATA box between Drosophila and whiteflies, with Drosophila being the top sequence and the bottom 5 being the identified U6 from the whiteflies. The literature suggested we look about 400 base pairs upstream from the start of the gene being the +1. We did about 500 base pairs to be generous. The Berkeley Drosophila Genome Project suggested that the TATA box would be 22 to 24 base pairs after the start of the gene, and that the proximal sequence element would be 13 base pairs after the TATA box. In conclusion, we identified with the help of the Berkeley Drosophila Genome Project that the whitefly's TATA box was 23 base pairs after the start of the gene, and that the proximal sequence element was 13 base pairs after the top box. In our next steps, we will test the function of the U6 promoters and find the optimal promoter for the small guide RNA expression, since there are 5 identified. In the future, we will put the small guide RNA directly into a plasmid and microinject in vivo. We will do this because DNA is much more stable than RNA, and there will be no need to reverse transcript once inside the plant, since the plant will already have a DNA template. And finally, we will test in a whole organism. I would like to give a big thank you to my PI, Dr. Linda Walling, for being so helpful and knowledgeable throughout my time with her. I started in a lab with her my first year as a lab assistant, and it's been great to have my responsibilities and knowledge grow. I would also like to give a big thanks to Alex Ensuela, a PhD student in the Walling Lab, who helped me immensely on this project. All of this wouldn't have been possible without Mr. Nee Tram, Ms. Jennifer Coplea, Mr. Jaron Patsky, Dr. Sue Wessler, Dr. Connie Nugent, Alex Cortez, and the entire RISE Peer Mentor staff. A big and final thank you to the National Science Foundation for funding this research. Any questions?
All right. Thank you very much, Stephen. Great presentation and a great ending as well. So if anybody happens to have any questions, yeah, please type in the chat. No one's taking advantage of the virtual hand here. Maya would like to know, what new skills did you learn for this research project? And great job, by the way.
Oh, thank you. So a lot of it was biotech skills, since my lab, the PhD student who I worked with did all this in the lab and then sent me his data. So I was trained on how to look at the base pairs in different softwares like Benchling and AdGen. So just learning all the different biotechnology aspects was definitely challenging, but a lot of fun and a good tool that I now have some grasp over using.
All right. And you stated since you were in the lab since the beginning of your career at UCR, how was that transition going to a remote setting?
It was definitely a little difficult because I really like doing like lab work and just being in person and communicating that way. So it's been a little difficult transitioning to online, especially since I work in like a botany and plant science lab. So it's very hands-on, but my lab has been really good about keeping us all connected and getting the proper training so we all know where we're at and we can continue forward.
All right, thank you very much. Great to hear that you're able to transition so well, and definitely great presentation again.
Thank you.
All right, we're gonna move along to our next presenter, Mr. Gabriel Chavez. Thank you for logging in. I see you're at UCR too, just like me. All right.
All right.
Your poster is up and running. Yeah, go ahead and you can have the floor.
Well, hello everyone. I'm Gabriel Chavez, and this summer I had the opportunity to work with Dr. Reddick in studying distance and seed preference in harvester ants. So foraging characteristics of harvester ants, they're known that they can change the landscape and vegetative populations of their environment. So how they do this? Well, harvester ants collect seeds, and when they collect seeds, they take them back to their nests and they eat them. So those seeds can no longer be used to grow and become a plant. So if we find what seed these ants prefer the most, we can infer that the vegetation, that the population of that species of plant will decrease and it will change the landscape of the environment. Now this is particularly important in California because the rough harvester ant, Pogonomyra tuberculosis, it resides in the coastal sage scrub ecosystem. If you don't know what this ecosystem looks like, think about the mountains behind UCR. That is coastal sage scrub. And this ecosystem is endangered due to fires, drought, urbanization, and invasive plant species. So finding out, investigating this rough harvester ant can give us greater insight into how to conserve the coastal sage scrub ecosystem. So what I did is I conducted cafeteria style experiments using invasive stinknet, invasive mustard, native brittle-bush, and native buckwheat seeds. I placed them at 3 meters away and 7 meters away to see if distance played a role in seed preference. So look at my methods now. So experiments took place at Mount Rimrock Reserve near Paris, California. I located 15 suitable nests for my experiments. So there were two 10-day experimental periods in beginning and end of August. During these periods, I had the Petri dishes here at my house, and I filled each dish with a certain type of seed, with 1 gram of the certain type of seed. The seeds I collected earlier in the year in June and July at Mont Rimrock. So when the seeds were all ready, I took them out the next day around 6 a.m. I placed them at each nest 3 meters away from the nest entrance. I left them there for 24 hours. I went the next day at around 6 a.m., picked up those dishes and placed new dishes at the 7-meter mark. Down here you can see how I placed the dishes. It's just a little diagram. The dishes were placed right in the center of their trail. I identified the trails and I placed them right in the center of the trail. I used a random sequence generator to find the order to place the dishes so that each nest, they were all random. I also conducted observations on select days at the 3-meter and 7-meter trials. Can we please click out of methods and head to our first graph? Yes. So here we have the average amount of seeds taken for each species of seed at each distance. So we see stinknet at 3 meters, it has the most. It was the most favored at 3 meters, followed by buckwheat, then burdobush and mustard. At 7 meters, we see buckwheat narrowly beat out stinknet, then it was followed by mustard and then burdobush. So something to notice here is the large drop-off in stinknet from 3 meters to 7 meters. So this tells us that the ants are choosing stinknet much more often closer to the nest than further. So why is that? Well, I didn't mention, but stinknet is the largest seed. It is the largest seed and it was predicted to be the most chosen because it is the largest. However, it couldn't be too large that the ants find it cumbersome to take it back to their nest. So nevertheless, there's something particular about stinknet that the ants favor because it was chosen most at the 3-meter distance. Can I please click out and head to the next graph? Thank you. So here we have the amount of seeds taken per species for the first 5 trials to the last 5 trials. As I mentioned, the first 5 trials happened at the beginning of August and the last 5 happened at the end. So we look at stinknet and buckwheat, and the favor remained almost the same, so it didn't change as the month progressed. However, if we look at mustard and burdobush, there's a drop-off. So this tells us that the ants were choosing mustard and burdobush less often at the end of August compared to the beginning of August. So why can that be? Well, buckwheat, it could be because the onset of buckwheat. So in the beginning of August, buckwheat was starting to go into seed, and it was much more prominent at the end of August. When you go out to the reserve, it's all beautiful red bushes, and that could be the reason why the ants were choosing mustard and burdobush very little compared to the beginning. If we could click out and head to my observations. So I went out 4 days at the 3-meter trials and 4 days for 7-meter trials. And I watched every nest for about 2 minutes and just to notice the behavior, what they're doing with the dishes and what they're doing with the seeds. And I noticed that the ants would take the seeds out the dish and they'll place them somewhere along the trail, either in a designated location, off to the side, in the front or back. And this was most prominent with stinknet. So they would take stinknet out the dish and they would wrestle around with it for a little bit and then they'll just leave it. So like I said, it could be because stinknet is so big, they realize maybe it's not worth it to take it all the way back to the nest. I don't know. With the other seeds, however, they would take it out the dish, they'll place it around the dish, make a little pile, then they would choose from that pile and take it back to the nest. And I found that very interesting. We could click out the observations now. So with my graphs, we ran it through data analysis in RStudio, and we found that species, distance, and time are all significant factors in the seed preference in harvester ants. So this data can be used to further investigate harvester ants and how they, the relationship with their environment, how they could change their landscape, and most importantly, how they can help conserve the coastal sage-grouse ecosystem that is endangered in California. Personally, for future projects, I would want to investigate the relationship with stinknet, how they harvest it, how they use it. I'd also love to recreate this experiment in a controlled lab setting. So, you know, there's no room for error. Before I finish my presentation, I'd like to thank Dr. Reddick. He took me under his wing about a year ago, and I've been a helper, and now I'm here presenting my own project. Also, graduate student Rebecca Keem, I couldn't have done this without her. She helped me every step of the way and made sure my project was doing well. Also, the LC CNAS scholars, they're great, and the alumni, specifically Mr. Kenneth and Nancy Gray, for the donations and making my project possible. Lastly, the RISE coordinators, you guys put on an awesome program, and my peer mentors for giving the best advice. That concludes my presentation. Thank you for listening. I will now take some questions.
Thank you very much, Gabriel. Great presentation. I was actually going to state that technically Dr. Reddick took you under his proverbial leg since you are in the entomology lab, and now you are a worker ant for him. So let's see, does anybody have any questions? Looks like you do have one. Do you weigh each seed, and if so, how much larger was StinkNet than the other seeds?
I do have the weights. I do not have them with me specifically at this moment, but StinkNet was substantially larger. I don't know the specific, I don't want to give a wrong estimate, but it was significantly larger. They're little bulbs. I don't know if you've ever seen StinkNet, but they're little bulbs, and they're very invasive to California also.
All right, and one last question from Dr. Purcell. Do you think the harvester ants might actually increase the StinkNet population?
Unfortunately, the way that this project, went about and the conclusions that I drawn, StinkNet is very popular with them, and they could increase, it could decrease. We have to continue to do more observations and more experimental data.
All right. Thank you very much. Looks like we went over a little bit on time, so we're gonna push right on over to our next presenters. It's actually gonna be co-presented by Ms. Stephanie Loyola as well as Shakira Palmer. Can both of you start your videos if you are able to? And your posters are up and running. All right, I will go ahead and duck out and give you guys the floor. Take it away.
Thank you. Good morning, everybody. My name is Stephanie Loyola.
And my name is Shakira Palmer. This summer, we're given the privilege of working under our PI, Dr. Jessica Purcell, and grad students Madison Sonkowitz and Mari West in the entomology department.
Today, we're going to be sharing with you our research into the world of ants, more specifically into the homes of ants. For a quick overview, ants perform many tasks in order to improve the chance of survival for their colony. One of those tasks is constructing a complex array of tunnels in the soil called a nest. Since ant nests provide essential protection, storage, and breeding space for a colony, they must be constructed to effectively provide these things in varying environments. So we ask the question, how does the environment affect nest structure? To answer this question, 100 Formica podzillica ants were collected from Boulder County from elevation spanning between 7,000 and 10,000 feet. They were housed in clear containers, and the containers were placed in different temperature treatments. These ants then excavated and created their nests over a week-long period, and photos were taken each day of the progression. Marissa, could you zoom into the materials and methods? Thank you. For the materials and methods, we analyzed the photos and collected data through the use of an application called ImageJ. We focused on two properties of the ant nests, area and depth. With ImageJ, the image was altered so that the tunnels that the ants dug could be recorded. The tunnels were highlighted in red, and by dragging a rectangle tool over the highlighted nest section, the area was able to be measured. We can see an example of this in the image on the screen. It's the first image that you guys see with the red. What you're looking at is, in the red is the highlighted tunnel that I mentioned before. For the nest depth, we used the line tool, and by clicking the lowest point on the tunnel and dragging it to the top of the dirt, we were able to determine a length. And we can see an example of that in the bottom image. Finally, for the statistical analysis, we used a Welch 2 sample t-test.
Now for the results. After gathering all of our data, displaying the depth and area for various elevations and temperatures, we determined the vast majority of our data did not have any statistical significance. This was due to the lack of a common trend between the two graphed values. However, we were fortunate to record statistical significance with our depth versus temperature graph. We determined that although the ants continued to excavate the same average amount of soil under warmer treatment, they always dug deeper tunnels. With this being said, it can be concluded that ants have a common trait of altering the depth of their nests depending on the temperature they're in. In conclusion, we found that there is not a significant difference in the nest structure throughout varying elevations. Oh, Marissa, can you go to my conclusion, please? Thank you. It was also seen that ants also excavated around the same amount of soil across varying treatments. These findings suggest that colonies of the same size can excavate around the same amount of soil. However, they do shift their nest structure to reach deeper into the soil when confronted with warmer temperatures. This conclusion tells us that nest structure is not a genetic adaptation of Formica podzolica. Instead, it is a phenotypic trait that can change with environmental conditions. So what's next? The next step of our project is to measure the branch nodes and tunnels. With this data, we will be able to see the complexity of the nest structure and determine how it varies under different elevations and temperatures.
We would like to take some time to thank the people that made this research possible. This project was sponsored by the Neil Allison Campbell Endowed Research Award and Miss Rochelle Campbell, as well as Dr. Purcell's 6-leg program funded by the U.S. Department of Agriculture and the National Institute for Food and Agriculture. We want to offer a special thank you to our PI, Dr. Jessica Purcell, and grad students Mari West and Madison Sinkovitz for all of their guidance for the opportunity to work alongside them. We also would like to thank our lab partner Zara Saeed for the support she showed us as our peer. And lastly, we'd like to thank Nhi Tran, Marissa Carranza, and all of the peer mentors, as well as everyone that made RISE possible. This would not be possible without all of your support. Thank you to everyone for listening, and we're going to open it up to questions.
Okay, thank you very much, ladies. Great presentation and great teamwork as well. All right, looks like we have, you do have a question from Ms. Maya. What was your favorite part about doing this research project?
Yeah, I'll go first for that one. So my favorite part was looking at all the ant data and just learning more about the ants, because we don't usually think about what ants do and why their tunnels and why their nests look the way they do. So it's definitely more interesting for me to go outside now and look at ants and then think about, oh, the ants are going to bring food to their queen and they're going to build a tunnel. And then to try to imagine what the nest might look like in our temperature and our elevation. So it just enlightened me in that way. And I really enjoyed that.
Yeah, I'll answer next. I also really enjoyed just pretty much learning more about the ants themselves, because again, you don't think about it, and seeing how they the progression from their structure being built. It was just very interesting to collect that data and watch them kind of progress in their nest structures. At first it's just a blank plate of soil, and then all of a sudden it's these very intricate nests. So that was very interesting to see.
All right, Marissa has a last question for you. How much deeper did the ants dig in increased temperatures?
So we don't have an exact number for that. We just looked at the overall data, and the data showed us that when the temperature was elevated and warmer temperatures, they just dug deeper.
All right. Well, thank you very much, you two. Great presentation. And we will be moving on to our last presentation of the day, Mr. Joshua Bocanegra. And you can go ahead and turn on your camera.
Marissa will have to turn it on.
Yep. I'll click that button for you. There you go.
All right, perfect.
Thank you very much, sir.
Hello.
Well, your poster's up. We hear you perfectly well. And you can take it away.
My name is Joshua Bocanegra, as Nhi introduced, and for the past 8 weeks, I've been working in Dr. Barsaka's lab out of the Physical Sciences Department of Physics. And what I've been trying to do is develop superconducting microwave resonators for quantum spintronics. Unfortunately, this is going to have to be more of an update than me presenting findings due to some manufacturing delays. And I'll get into that more in the meantime, but I'll explain what it is that we were doing. We would like to take a superconducting material, superconducting resonator, and couple it to a YIG, yttrium iron garnet, in order to observe magnon spintronic effects within the material. Now, what we wanted to start with was building the superconducting resonator, right? The goal of the project was to start with that and go on from there. But before we even got to superconducting, we wanted to start with our just regular conducting material, just create resonators out of gold and copper and see how well those did in order to continue on and develop the superconducting part afterwards. Now, why superconducting? Superconducting allows us to do the very superconducting materials have zero resistance. And so when we work with them, we are able to see much longer lived signals in electrical impulses. And we are able to get much, much finer detail in our measurements. If you can click on the first image at the bottom, figure 1, the timeline of superconducting materials. We wanted to work with something new, and this is all, as you can see, the graph starts in the 1900s, but in 1985, there's 1980s, there's a big jump, and we see the first time what they call high temperature superconducting materials with yttrium, barium, copper oxide up at the top, and it's formerly known, or currently more known as YBCO, and we would like to use this material in order to develop these resonators. Can you click out and then click on the YBCO page? The reason we'd like to use YBCO is because instead of having to go all the way down to liquid helium temperatures, which are around 4.1 Kelvin, we'd have to go instead to 77 K to use liquid nitrogen. And this allows it to be much more economical for us and much more easier to work with. The cryogenic temperatures, the methods that we have to use are much less severe and easier to go along with. Can we back out now? And then go on to the manufacturing of the resonators. And this is what I've been doing for most of the presentation. That's not the picture up at the top right. Well, we can just stay there, that's fine, that's fine. Yeah, okay. So creating the DXF files was the first thing I did. And the reason I had to do this, we had to use Python libraries. We had to create the designs that you just saw previously for a second. And we had to make those, send them out to get shipped by Martin Photomask. And that took a while. And then we had to grow the test samples with gold and plasma sputter. And this is where we had our issues. Continuing on though, we plan to measure and test the samples, then fabricate YBCO, and create the superconducting analogs after we have tested the gold samples first, and then continue on from there. Now, we have already ordered the YBCO prefab samples, and so I've had to do a lot of research deciding how we're going to process them, how we're going to make them, and how we're going to continue to work with the material. YBCO is a ceramic. It's actually made of, as I said it is, but there's a layer of copper, a vertical layer atomic copper, and then there's a layer of barium, and then there's a horizontal layer of copper again, and then in the middle there is the yttrium, and then it's mirrored on top of each other to create a very, very complex structure. And so it takes a while to make that, and it's very expensive to produce that. So we produced simple centimeter by centimeter samples that are on their way. And once we have tested and grown the metal, the gold samples first, we will continue on to manufacture the YBCO and then move on from there. Once we do that, we will be able to couple that with the previous work that Dr. Barsa Barsukov has done in his lab and be able to make very, very sophisticated quantum measurement devices of the magnon. And if you can click on that image there, figure 2, these are some of the samples that we made to test. So we'll use a photolithography technique to grow the samples of the gold and then measure them from here. And we used a very simple linear approximation for our first findings. We had hoped to have these done and printed to have some data to show, but due to one of the grad students in the lab having to take some personal time and then some of the shared fabrication space within the Department of Physics, there were some issues with our evaporator and our plasma sputter that prevented us from growing it. And so we'll have to continue this. And so that's where we're at right now, just waiting to get those samples finally finished, grown, and then test it and move on from there. And so if you click back out, the next steps that we'll have, I don't have a slide to zoom in on, but the next steps, I will have to use ANSYS software to do some modeling of our devices to get more high fidelity, to iterate over what we've already done. And then from there, go on. And then we'll be looking at whether or not we want to stick with the YBCO, because the YBCO itself is just a proof of concept. If we can do it, then we'll have to decide if we want to continue from YBCO and on to niobium, or how we're going to do that. And there are some issues with YBCO because it's so complex. It's, like I mentioned, it's hard to fabricate, but it's also very easily corroded by water. So we had to passivate the layer with a layer of silicon dioxide when we fabricate. And so there are just a lot of fabrication methods that we have to consider and continue to watch out for as we do it. And then hopefully within the fall, we'll also be able to start beginning coupling the YBCO resonators to the YIG magnets and for measurements. And at that time, I'd like to open it up for questions. And if you have any questions at all, I'd be happy to answer them.
All right, thank you very much, Joshua. Great way to close it out. If anybody happens to have any questions, please feel free, type in the webinar chat here. It looks like we do have one virtual hand. So
Sure.
Devante,
Okay.
I will unmute you and you can actually ask Joshua your question directly.
Hello, can you hear me?
Yes, I can.
I wanted to know, for your manufacturing process, what was the most sensitive or most important variable that comes into the cost of the YBCO of making this product?
So if you remember the money that the program provided for us at the beginning of the year, that was gone within the first 2 weeks. Just manufacturing the photomask that you see there in the bottom, that disappeared immediately. To actually produce 4 little tiny centimeter by centimeter square prefabs of YBCO cost over $2,000. And that doesn't include any of the other additional manufacturing that we're going to have to do ourselves. Much of the research done was looking at how to continue with that, right? And see how we were going to do it in lab, as well as reaching out to a bunch of other people to see, what their capabilities, what the industry capabilities were. And I forgot to make my acknowledgements. I'd really like to do that this time. I'd really like to thank Dr. Barsukov, Dr. Brandon Brown, Nitran, Marissa Carranza, Basim R. Cook, Sharash Rejmi, Rodolfo Rodriguez, Eric Hagen, Elizabeth Williams, the Barsukov group in general, and then Star Cryo-Electronics, Martin Photonics, and then Gomtech Electronics were very helpful getting specifics of research down. There's a lot of manufacturing specifically that only those people can do that I had to talk with them and learn about. I would say about 30% of my time, it was split equally between actually like troubleshooting software. There was some huge issues with that, researching like manufacturing techniques, and then actually doing work for this whole thing just because it was such a big project. But yeah.
All right, thank you very much, Joshua. That actually brings to a conclusion the second day, day 2 of our RISE Symposium. Thank you everybody for attending, and tomorrow at the same time, same channel, we will host the 3rd and final RISE presentation with the remaining presenters. As always, we will begin with some opening remarks. We have the last introduction video, so we get to see everybody all the remaining presenters and their elements. And we have a few less presenters, so we will be ending at approximately 12:20 tomorrow. And you can have an early weekend. But again, thank you very much, everybody. Great seeing you all. And we will provide this presentation as a recording as soon as we receive it. Have a great lunch because it is lunchtime. Thank you, Dr. Nugent. Mentioning everybody did a fantastic job, and we will see you all back tomorrow. Goodbye.